Iwanan mo na yung mga dito.
All-powerful God, you are present in the whole universe and in the smallest of your creatures. You embrace with your tenderness all that exists. Pour out upon us the power of your love, that we may protect life and beauty. Fill us with peace, that we may live as brothers and sisters, harming no one. O God of the poor, help us to rescue the abandoned and forgotten of this earth, so precious in your eyes. Bring healing to our lives, that we may protect the world and not prey on it. That we may sow beauty, not pollution and destruction. Touch the hearts of those who look only for gain at the expense of the poor in the earth. Teach us to discover the worth of each thing, be filled with awe and contemplation, to recognize that we are profoundly united with every creature as we journey towards your infinite light. We thank you for being with us each day. Encourage us, we pray, in our struggle for justice, love, and peace. Our Lasallian prayer. I will continue, O my God, to do all my actions for the love of you. Saint John Baptist de La Salle, pray for us. Live Jesus in our hearts forever. Amen.
Good morning, everyone. Welcome to Trends and Approaches in Analytical Sciences 3 or TAAS 3, the Metabolomics Approach and its different applications. So sorry for the long wait. I am Chris Argamino of the DLSU Central Instrumentation Facility or CIF, and I will be your host for today. So, chika muna tayo. Have you had breakfast and coffee already? So kung hindi pa, sabayan namin kayo sa kape at almusal. Medyo mahirap gumising ng maaga pag weekend, but we're really thankful for your presence here. We assure you that this morning will be very fruitful and productive as we try to unravel the mystery of metabolomics in this webinar series. So first, before we officially begin the event, may I remind everyone of the general privacy notice and house rules for the entire webinar session today. So as you have seen dun sa poster natin, we have 3 sessions, March 6, March 13, and March 19. So this is applicable to all sessions. First, screenshots and video recordings of the event will be done and may be used for print, online, and social media publications. Number 2, this event may include media coverage and story releases online, in print, or on traditional broadcast channels. Information collected via registration or media capture may be kept by the CIF for archival purposes in the Google Drive account. Number 4, personal recording of the session shall not be done to respect the rights of participants in the session. Fifth, the recording of the session shall not be reshared and or reposted online without prior notice from the organizer to prevent unauthorized disclosure and processing of personal data. And the last one, for the general privacy notice, discussions and conversations of a personal nature not related to the ongoing session shall be discouraged to prevent unintentional and or unauthorized disclosure of personal data. We now move on to the house rules. So for our house rules, first, this webinar is being recorded and you will be able to view it on demand later via the live section of our Facebook page. At any point in the webinar, feel free to type in your questions via the Q&A box for Zoom and via the chat box for our Facebook live stream participants. Third, the speakers will try to address as many questions as possible during the open forum later. If time does not permit, our team will get back to you with answers via email. Lastly, e-certificates can be requested by participants who have both pre-registered to the webinar and have submitted the webinar evaluation form after each session. So as of earlier this morning, we have 600 plus participants who have registered. The link to the WEF will be posted later in the Q&A box, FB live chat box, and CIF page. The WEF can be submitted until tonight, 11:59 p.m. So konting recap muna tayo. So this is not the first TAAS, so this is the third one. And as some of you may be familiar, a year ago, we held the inaugural Trends and Approaches in Analytical Sciences or TAAS 1 at the Yuchenko Hall of DLSU Taft. Medyo swerte tayo nun kasi this was done before the ECQ or the lockdown. And this was attended by hundreds of participants from various sectors. So in TAAS 1, we learned about modern trends in laboratory instrumentation from our guest speakers from two suppliers in the Philippines, DKSH Philippines and Shimadzu Philippines. Our keynote speaker then was DOST PhD, Balik scientist Dr. Lionel Santos, who shared his expertise in TAAS 1 on pharmaceutical impurity analysis. So also last year, we weren't able to do a physical session because of the pandemic, but we're able to hold the second TAAS, which was held in webinar format. So for that event we once again had the US-based Dr. Santos. And in TAAS 2, he was able to share his expertise naman on the various topics associated with method validation in the laboratory. So for this third installation of TAAS, we are lucky to have two speakers. The first one is Dr. Ana Karen Lacerna, and she is a DOST PCHRD Balik scientist who will be presenting on the introduction to Metabolomics. And our guest speaker is Dr. Maria Fe Kalingason of the DLSU Chemistry Department, who will be presenting on plant metabolomics. So before heading to the webinar proper, let us hear some welcome remarks and the introduction to the CAF from our director, Dr. Drexel Camacho. Sir Drex.
Good morning, everyone. It is with great pleasure that we welcome you today to the third installment of the Trends and Approaches in Analytical Sciences 3 or TAAS 3. At the same time last year, as discussed by Chris, just before the lockdown, we held the first TAAS featuring Dr. Lionel Santos, the Balik scientist we hosted under the DOSTP Shield. This was followed by TAAS 2, which was done online. Today, we are fortunate to host another Balik scientist, Dr. Ana Karen Lacerna, this time under DOST PCHRD. Metabolomics is a very powerful field that uses analytical data to analyze components in a way that gives us the underlying differences between chemical components in relation to some factors. It is a very interesting field that can be applied to many different applications. Hence, we deemed it proper to hold this webinar in 3 installments. Today, Dr. Lacerna will introduce the basic principles of metabolomics, followed by its application in plant metabolomics to be presented by Dr. Maria Fe Kalingacion. Next Saturday, March 13, Dr. Lacerna will continue with the metabolomics presentation, followed by its application in food metabolomics. Then on March 19, Friday, we continue exploring its applications in clinical metabolomics. We hope that you will learn a lot from all these sessions, and we hope to ignite your interest to pursue research in this field. So we at the DLSU Central Instrumentation Facility are a willing and able partner to your research needs. So let me take this opportunity to introduce to you the De La Salle University Central Instrumentation Facility. The facility is located at the DLSU Laguna campus in Binyan, Laguna, and it is the premier instrumentation center of De La Salle University. It is under the Office of the Vice Chancellor for Research and Innovation, and it aims to elevate the quality of scientific research, not only in DLSU, but also in the entire country. So the team, or the CIF, is manned by the following team, composed of faculty, equipment specialists, and dedicated laboratory analysts. Let me introduce to you the laboratory equipments that are available in our center that will help you in your research needs. First, we have the sample preparation laboratory equipped with the common laboratory instruments that will help you process your sample, the microcentrifuge, semi-microbalance, freeze dryers, sonicator bath, water purification system, ultrasonicator probes, among others. Then the NMR laboratory is equipped with the most advanced NMR instrument in the country. It's 600 megahertz NMR instrument. It has a very powerful magnet, superconducting magnet, and it has a 2-channel amplifier system capable of auto-shimming, on deuterium digital lock system, and as an auto-sampler. The NMR machine is equipped with a liquid probe, which allows us to analyze sample in solution in both 3 mm and 5 mm NMR tubes, and it is also equipped with a solid probe, which allows us to analyze sample in solid form. We offer services like the routine NMR analysis, 2D NMR analysis, multi-nuclear NMR analysis, multi-pulse experiments. We also do variable temperature experiments in the range -100 to 150. We do traditional inorganic multi-nuclear analysis, and we can do solid NMR analysis as well as copolymer analysis. We also offer trainings for basic NMR spectroscopy for chemistry and non-chemistry majors. We also offer training for structure elucidation using NMR spectroscopy, as well as advanced techniques in NMR spectroscopy. The spectroscopy laboratory is equipped with instruments used for spectroscopy. So the Shimadzu UV-Vis spectrometer is equipped with a reflectance attachment, which allows us to analyze not only solid solution, but liquid solution, but also solid samples. We can analyze reflectance because it's equipped with integrating sphere attachment, allowing us to analyze sample in solid state. Small and large samples are possible. We also have the FTIR equipped with ATR attachments, which allows us to analyze samples directly into the instrument. We also have the Malvern synthesizer, which allows us to analyze the particle size of nanomaterials, as well as zeta potential and the microreology of the solution, particularly useful in proteins and electrophoretic mobility of proteins and polymer solutions. We also have an ICP-MS, which allows simultaneous analysis of multiple elements, and we can analyze trace levels at the ppb level. The elemental analyzer, which allows us to determine the percent composition of carbon, hydrogen, nitrogen, sulfur, and or the presence of chlorine. And this one is useful for characterizing organic compounds. The Chromatography Laboratory is equipped with the following instruments. The HPLC, which is capable of converting into a GPC or size exclusion chromatography. It is also equipped with the UV and RI detector. And we also have a preparative LC system with fraction collector, which allows us to collect samples from separated components in the LC system. And we also have the LCMS QTOF, okay, which is useful in metabolomic studies as well. And of course, the GCMS, which allows us to monitor components in volatile components in a sample, which is again useful for metabolomic studies. The Integrated Electron Microscopy Laboratory is equipped with the following instruments. The Transmission Electron Microscope, or the TEM. So we have an ultra-high TEM, which can do resolution as high as 0.19 nanometer. It is also equipped with energy dispersive spectrometer for elemental analysis, and it can do elemental scanning as well. And along with the TEM, we have an ultra microtome instrument that allows us to thinly slice sample for measuring or for viewing in TEM or SEM. And we have a micro manipulator so that we can prepare samples in the nanoscale. And of course, we have the palladium gold spatter coater, which is useful for preparing the sample. The TEM is capable of analyzing nanomaterials as well as biological samples. We also have the scanning electron microscope, which is also equipped with an energy dispersive EDS, and then it allows us to do elemental mapping as well. Then we have the focus ion beam, which allows us to manage your sample or operate your sample by milling and polishing your nanomaterials, and we can do that in focus ion beam instrument. Upcoming is the Imaging and Cell Culture Laboratory, which is ongoing installation now. And we will be having this instrument called the confocal laser microscope with live cell imaging. So this allows us to visualize cells live, and we can monitor the interaction of a compound in interaction with the cell. And this one is also coupled with cell culture laboratories, which allows us to prepare samples from plant cells, mammalian cells, as well as bacterial cells. And of course, for engineering research, we have a wind tunnel facility, which allows us to study aerodynamics and as well as modeling of air flows. So the Central Instrumentation Facility offers programs and projects, and the following. The first one is we offer graduate residency program where we invite graduate students to reside on campus so that they can have access to the instruments 24/7. And we recently graduated two students for this program who did 24/7 access to the instruments and unlimited use of the instruments. Okay, so we had Marielle Texon, who finished her MS degree last term. Next, and we have Jessa Luteri, who also finished her MS degree last term. And ongoing now, our research resident is Paul Adrian Go, who is ongoing doing his thesis, okay, on campus. And we also have the Plus Lunas Fellowship Program, which is funded by PCHRD, where we invite fellows all over the Philippines from different Plus Lunas centers. And we had a phase 1 training, which is online, training on chemistry of natural products, NMR spectroscopy for natural products, and chromatography for natural products. So basically, this phase 1 or online session will be followed by phase 2, wherein all these fellows all over the Philippines will be brought to De La Salle University in Laguna campus for hands-on operation of these machines. Then we host Balik Scientists. The first one, as mentioned earlier, was Dr. Leonel Santos. And it was very instrumental in ensuring that our processes are at par with international standards. And of course, right now we are hosting Dr. Ana Karen Lacerna, who will be our keynote speaker for today's session. And the services we offer at the Central Instrumentation Facility, we offer analytical services geared towards your specific objective in your research. We offer trainings upon request. We offer collaboration, and we also do consultation as well as research outsourcing. So for more information about the Central Instrumentation Facility, please contact our general email is cif@dlsu.edu.ph. We also have ongoing construction, the website that contains, so far at least now, the NMR facility. And you can contact us at Facebook and Messenger, just type DLSU Central Instrumentation Facility. So we hope that through the CIF and our activities, we will be able to help you in your research needs. And please do not hesitate to contact us for your specific objectives of your research. And we hope that we will be able to engage you and in detailed activities on your research. And we hope to elevate the quality and the number of research in the country. And let's help build our country through research. Thank you again, and God bless us all, and may you learn a lot through this 3-day seminar on TAAS 3. God bless us all.
All right. Thank you, Sir Drex, for the very warm welcome. And we hope that the presentation piqued your interest in the CIF's facilities and services. Again, for inquiries, you may send a message via our Facebook page, DLSU Central Instrumentation Facility, or you may send us an email via cif@dlsu.edu.ph. Now, we will be introducing our keynote speaker. Okay. So our keynote speaker is a Department of Science and Technology Philippine Council for Health Research and Development Balik Scientist awardee. She earned her Bachelor of Science degree in Chemistry from the University of the Philippines, Los Baños, and spent her early professional career as a lab analyst at the Analytical Service Lab of UPLB Institute of Chemistry, then as a Quality Assurance Officer at Me Johnson Nutrition. She then spent the next decade in NUS, the National University of Singapore, as a PhD in chemistry student, research assistant, and as a postdoctoral research fellow, covering a number of projects involving various applications of metabolomics. In addition, our analytical science projects in areas such as preclinical/clinical, agriculture, food, forensic science, and environmental research. In her ongoing tenure as Balik Scientist, she has assisted the CIF through her technical support in the daily operations of the Chromatography Lab, and her involvement as a mentor for the Chromatography Lab course of the Tuklas Lunas Fellowship Program for Natural Products Phase 1, a project funded by the DOST PCHRD. Our speaker has also served as a lecturer at the DLSU Chemistry Department, handling the graduate course on special topics in analytical chemistry, specifically food analysis, in Term 2 of AY 2020-2021. the Santa Rosa, Laguna native, a rising research scientist in the field of metabolomics, has published more than 10 papers on the topic covering various applications. Everyone, we are honored to introduce you to our keynote speaker, Dr. Ana Karen Lacerna.
Okay, so thank you, Chris, for the introductions. Good morning, everyone. So I'm Karen Lacerna, and today I will be presenting on the introduction to the Metabolomics approach and its applications. So this is the first part of the three-part webinar series that we have on metabolomics. And for this webinar series, we'll try to answer your questions regarding metabolomics. So we'll try to cover what is metabolomics, why should you consider using it in your research, when and where can it be applied, or in what areas of research can it be used for, and lastly, we'll cover the how, or how can we perform a metabolomics experiment. So for this session, we'll cover more of the what, the why, the when, and the where. In the subsequent sessions, I will be talking more about the hows of metabolomics. So we'll cover the experimental and the practical considerations when performing a metabolomics experiment on March 13. And we'll cover more of the data analysis aspects of metabolomics on March 19, which is basically on how do you use chemometrics in metabolomics data analysis. So first, we cover what is metabolomics. So yung metabolomics, actually, mahati mo yung term into two different words. So you have metabolome or metabolite, metabolism, and omics. So first, we talk about metabolism and metabolites. So metabolites refers to the totality of the cellular activity. And as you can see from this diagram, it is a highly coordinated network. And this is also actually very highly regulated. So it consists of different reactions of chemicals, which we'll talk more about later. And this network consists of smaller systems or multi-enzyme systems that are known as the metabolic pathways. So these metabolic pathways can perform different functions. So it can be for purposes of generating energy. It can be for conversion of nutrients to endogenous molecules or molecules that you find in the body. It can be to build larger molecules from the monomers or the building blocks. And it can be to break down toxic substances as well to metabolites or compounds that can actually be excreted from the body. So just now I have already mentioned metabolites. So metabolites can, in this case, makes up your metabolic pathways. So they are the substrates and the products, the intermediates of the metabolic pathways. And these metabolites are actually small molecules that are produced in or by biological processes. So they are in the molecular weight ranges of 50 to 2,000 Dalton, and they can be endogenous. So when we say endogenous, these are metabolites that are produced within the biological system. So this can be primary metabolites. So primary metabolites referring to those that are part of the primary metabolism of the biological system, such as your amino acids, your carbohydrates, your lipids, organic acids, etc. In the case of some other organisms like your plants, you will have your secondary metabolites, which are actually metabolites that are produced in response to either stress or as a part of their defense mechanism. You also have exogenous metabolites. So exogenous metabolites naman are those, so if you have synobiotics, which are like foreign agents of the body or drugs, they are also metabolized by the biological system. And these are what you know, or what is called as exogenous metabolites. So here are just some examples of these metabolites. So you have phenylalanine, which is an amino acid. You have glucose 6-phosphate, which is a phosphorylated version of glucose, a well-known molecule for most of us. And strychnine is, of course, an alkaloid, a secondary metabolite. And here also is acetaminophen glucuronide, which is acetaminophen, your paracetamol, that is metabolized into the glucuronide form. So, the next part of the metabolomics term is your omics. So, kapag sinabi natin bang omics, ano ba yung omics? So, when omics is used, it is used to reflect totality. It is used to reflect the use of high throughput technologies in order to simultaneously and comprehensively measure certain molecules. So, depende na lang dun sa kung ano ba yung nasa first part of the term. So, for example, when we say genomics, it is to simultaneously and comprehensively measure genes of a particular biological system. When we say transcriptomics, it will be to measure transcripts, gene expression. When we talk about proteomics, it will be to measure proteins. Glycomics will be to measure carbohydrates. Metabolomics, as what we're going to cover now, is to measure metabolites, and lipidomics is to measure lipids. So as you can see here, from the cascade, from the molecular cascade here, you have your from the chromosomes, you have your genomic expression, and down closer towards the phenotypic expressions, you have more of the smaller molecules such as your lipids, metabolome, or the metabolites. And in this part, glycomics will be more covering on the post-translational modifications of your proteins wherein they can be glycosylated. So yun naman yung sa part na glycomics. So, metabolomics. When the term was first coined in 1998 by Oliver et al. When they measured the changes in metabolite concentrations with deletion or overexpression of a gene. So they were looking at the changes in the metabolite concentration with the deletion and overexpression of genes in yeast experimental model. The term metabolomics, on the other hand, was first coined by Oliver Finn in 2002 when he talks about metabolomics as being the comprehensive analysis wherein all metabolites in a given biological system are identified and quantified. Now, another group also came up with the term metabonomics. So this is actually coined by Nicholson et al. In 1999, and this is defined as the comprehensive and simultaneous systematic profiling of multiple metabolite levels and their systematic and temporal changes caused by factors such as diet, lifestyle, environment, genetic effects, and pharmaceutical effects, both beneficial and adverse in whole organisms. So medyo mas comprehensive yung definition ng metabonomics. And in some cases, during the early parts of this metabolomics era, medyo interchange siya, and in some cases some would say that okay, metabonomics is subdivision of metabolomics. On the other hand, yung iba naman sinasabi that metabolomics is subdivision of metabonomics. So kumbaga, yun, there is a lot of contribution in the terms, but eventually ngayon it's more of metabolomics that is being used. And just to sum everything up, when we say now metabolomics, it basically refers to the simultaneous and comprehensive analysis of metabolites in a given biological system under study. So, in order to do metabolomics, you need to make use of certain instrumentations, and these are some of the main instrumentations that are being used, particularly more of the MS-based and the NMR instrumentation. So, you have the spectroscopic techniques or spectroscopic instrumentation, mainly used for purposes of fingerprinting, whereas MS-based and the NMR spectroscopy, which actually can allow you to identify metabolites, are more for the different purposes. So MS-based metabolomics can make use of either direct injection or coupled with separation techniques such as liquid chromatography, gas chromatography, as well as capillary electrophoresis. So we'll talk more about these instrumental aspects when we go into the experimental considerations and workflows of metabolomics in our next session next week. So why should we take the metabolomics approach? So we have your genomics and proteomics, which are the more established and older levels of omics approaches. So now, why should we consider using metabolomics? So first, we have to consider yung similarity of metabolites across different species. So just think about it. For genes and proteins, there can be differences. There's a lot of diversity between genes and proteins across the different organisms. Whereas for metabolites, glucose, for example, in pigs is glucose also in humans or in other organisms, like may provide a different function in different organisms. So there can be diversity in that. Whereas for metabolites, it is highly conserved. You can also consider yung proximity niya to the phenotypic expression. So not all genes and proteins get translated to a phenotype. So when we talk about phenotype, this is yung actual manifestation of the biological system, kung ano yung nakita mo, kung ano yung state of the biological system that it can be observed. And yun nga, so in genes and proteins, not all gene and protein expressions are translated into the phenotype, basically because you have post-transcriptional modifications and post-translational modifications that occurs, and these are not captured kasi yung mga yon is nangyayari after ng gene expression and after the protein expression. So these post-transcriptional and post-translational modifications, as well as environmental influences, are actually captured by the metabolites or in the metabolomic expression. So metabolome is closer to the phenotype. You can also expect that from the gene expression, there is also a magnification of the changes as it goes to the metabolite level, and this provides increased sensitivity in terms of like detections. Metabolome can also reflect effects of environmental factors, health condition, diet, gut microflora, host interactions. So lahat yun nakakapture in the metabolome, and this can provide you with a better, in a sense, more accurate representation of your phenotype in terms of the molecular aspects. And lastly, it has easier translation to diagnostic and monitoring devices and assays. So I'm sure a lot of you or all of us are familiar with the use of glucose. So glucose as indicator or a biomarker of the development of diabetes, for example, and you also have your creatinine measurements that serves as a physiological monitor for the development of renal dysfunction. So alam natin na kapag medyo may problema yung kidney, tumataas yung creatinine, and kung merong diabetes, mataas yung sugar. So these are just a few examples of the use of metabolites to indicate disease or physiological condition. So meron ng utility. Even before the time of metabolomics, there is already the utility of metabolites for these purposes. So this is just to illustrate kung the numbers involved when we talk about the different omics levels. So in genomics, you have about 40,000 genes. Transcriptome, you are dealing with around 150,000 different transcripts. Proteome, you have over or around 1 million proteins that are currently detected. And in terms of primary metabolism, you have about 7,800 metabolites, but this is of course not including secondary metabolites in plants and microbes, which is in the case of plants can reach up to about 200,000. So mostly in terms of the functions, mas known yung functions of the metabolites compared to like the level of your proteomes, transcript, and the different genes in terms of like effects of environmental influences, mas reflected din yung environmental influences in your metabolites. Now, this is of course not to say that metabolomics is the end all and be all among the omics levels. So this is definitely not the case. Actually, in the same way as you look at a map, so di ba yung mga maps natin, actually, if you look at Google Maps, you can actually divide a map into different layers. And you can consider or see your different omics levels as different layers of that map. And only in making use of the different layers are you able to see a better picture or a better view of the overall picture. So if you make use of this multiple layers, so mas maganda or mas comprehensive yung makocover mo na picture. And so usually, in order to better understand biological systems, there can be a need to make use of one, two, or more of these omics levels. And I think for the applications later, as well as in the applications that will be shown in the subsequent sessions, you will see this integration of different omics in different studies. Okay. A disease state, for example, or if you are looking at wild versus mutant plant species or knockout models of certain diseases. So you can use metabolomics to characterize them. It can be used to assess the effects of a treatment. So it can be, if you're trying to assess the effects of drugs, of certain pollutants, or in the case of like stress. So If you are looking at the stress response of, for example, plants, so you can use metabolomics to assess the effects of those treatments. It is also used to identify potential biomarkers. So when we talk about biomarkers, so an example again of this will be your glucose and creatinine, which are respectively used for the monitoring of the development of diabetes and renal dysfunction. So you can make use of metabolomics to characterize different disease, and from there on, try to identify metabolites that can be indicative or can be used for early diagnosis of these disease states. And lastly, it can be used for metabolite fingerprinting. So when we talk about our fingerprint, diba? So yung fingerprint, it is actually unique per person. So in the same way, like different organisms, different biological systems will have a unique fingerprint that can be obtained using its chemical composition. So in this case, we can look at the metabolite fingerprint of the individual, of the organism, and these can actually be used for purposes of authentication, establishing the origins of a particular organism or product, and it can also be used for purposes of quality control. Okay. So, where can we use the metabolomics? So, ito naman, we'll talk more about the actual research fields and areas and examples of the types of studies that metabolomics is actually used in these areas. So, this is by no means exhaustive. I'm sure you still have a lot of other research studies and research areas that you may be working on that are not included in the next slide. So if you see the potential of the use of metabolomics in your research, please do let me know if in any way I can be of help. And we can explore how to use metabolomics in your particular research area. Okay, so when some of these applications of metabolomics will be, of course, in plant biology, when looking at transgenic research, what are in terms of like changes of the metabolic profile of these transgenic modifications. It can also be used to characterize different breeds. It can also be used to study the mechanisms involved in plant pathogen interactions. It can be used to assess the stress response of plants, and it can be used as part of the functional genomic studies. In the In the case naman ng food and nutrition, it can be used to establish host microbiome interactions. Particularly if you look at the gut microbiome, so your gut microflora, yung mga microorganisms or microbes that you have in your gut also produces their own set of metabolites. And actually, this can also be reflected in our own metabolic profiles. So, and in addition to that, pwede mo tingnan yung effects of changes in the gut microbiome and how this affects the metabolic profile of the host. It can also be used to personalize nutrition. So, dito po papasok yung mga current dito papasok yung mga current trends in terms of like personalization of, like, for example, medicine, personalization of nutrition, of diet in reference to like your particular genomic characteristics. So dito papasok yung mga nutrigenomics, the use of nutrigenomics, and together with that, you can make use of metabolomics to characterize your the persons or the different persons. So you can also use it for nutritional assessment. So nutritional assessment, because of course your endogenous metabolites are actually good measures of the nutritional needs of a particular person, especially in the case of studying malnutrition, and what are the needs for supplementation. It can also be used for purposes of authentication and quality assessment and control. So, dito papasok yung making use of metabolomics, metabolite fingerprinting, for purposes of authentication and quality control. In terms of natural products research, currently, as we have covered in our recent Tuklas Lunes program, fellowship program, you can make use of metabolomics in the replication in drug discovery. So, instead of going through the usual classical approach, one way or as complementary to the more established ways of doing drug discovery research, you can make use of metabolomics to identify, okay, in terms of like, you can correlate your assays. So ano ba yung mga different components or fractions that are bioactive, and you can make use of metabolomics to determine ano ba yung mga metabolites that are present in those fractions. It can also be used to identify the effects of these natural products in the person or in the organisms that consume them. So for example, your herbal products, traditional herbal medicines, what are their effects in the body? And this can be characterized using metabolomics. And again, making use of metabolomics in terms of metabolite fingerprinting, it can be used for purposes of quality control. Kasi as we know, natural products are very comprehensive in terms of like components, complex mixtures. So herbal products can make use of metabolomics approach to characterize or establish yung the profile of a good quality herbal product, and from there it can be used for purposes of quality control. And of course, it can also be used for authentication. So I think in some cases, particularly in traditional Chinese medicines, there can be adulterations, or especially if the contents are quite expensive, there can be cases of adulteration or total substitution of other products, and you can make use of metabolomics to characterize the metabolic profile of an authentic product. In the field of agriculture, you can make use of metabolomics for assessing crop improvement, for quality assessment of crops, for the assessment of growth conditions and treatment effects, as well as to establish geographic origins. So, for example, crops that are of the same variant that are grown in one area will have a different metabolic profile compared to crops that are grown in another area. So, you can make use of these metabolic profiles to distinguish ano ba yung produced in one area and what is produced in the other area. Sorry. Okay. So in terms naman of environmental research, metabolomics can be used for toxicological assessments and to in exposure studies to establish kung ano ba yung effects of these exposures and toxic compounds in the biological system. Lastly, we look at preclinical and clinical applications of metabolomics. So it can be used for, again, purposes of biomarkers, for physiologic monitoring. It can be for diagnostic biomarkers. So it can be used to diagnose the presence of certain diseases. It can also be used, for example, there are some metabolites that can be used to assess the effects of certain treatments. So maybe one of these could be your glucose. So if your diabetes treatment is working, then of course you can expect that your glucose levels can go down. In the same case for creatinine, if your kidney, renal drugs or kidney treatments are working, then you would also expect your creatinine levels to also go down. It is also very commonly used in order to study disease mechanisms. So metabolomics has been used in large cohort studies such as your Framingham heart study, as well as your in Europe they have this U-Biopred asthma study where they make use of metabolomics to characterize the molecular phenotype of asthma as well as cardiovascular disease. You also have your, it can also be used to identify drug targets in pharmacology to assess the effect of drugs and to establish whether safe ba to take these drugs. So in some cases kasi, actual manifestations of like problems or complications with the use of certain drugs can be coming much later. And hindi kagad siya makakapture in the physiological state or in the phenotype of the person. So it is good to make use of metabolomics to monitor these different metabolites kasi this can be indicative of like perturbations or bad effects of certain drugs. So kapag nakita natin that there are certain metabolites that are more associated with development of renal dysfunction or development of liver dysfunction, if you see that with the treatment of or with the use of certain drugs tumataas na yung mga metabolites na yun, then it is a time to consider whether to continue using yung drug or to see whether the dose can be lowered and so forth. So these are again different applications of metabolomics. So these are basically more of those that come to mind for in my case. But as I mentioned earlier, this is in no way exhaustive, and I'm sure you guys have a lot of different studies and use and can make use of metabolomics in your research. So please do let me know if there's any way that I can be of help and assist you in your research on how to make use of metabolomics. And so in the next part of this presentation, I will give you snippets of how I used or how our group has used metabolomics in different projects. So I'll give snippets of 2 projects that I have worked in collaboration with students as well as with collaborators in NUS. And also I will give more detailed presentation on an application that I have been particularly involved in terms of a preclinical model. So for the first application that I'm going to show, this is the work of a collaborator, Song Shuang and Srishti, together with their professors Wang Chiwa and Yu Tan. And the health services, HSA organization of Singapore, so Health Services Authority. So, sorry. So for this application, they made use of biochar for urban agriculture, and they wanted to assess the effects of how this biochar application can have on the bok choy. So bok choy or Brassica rapa is, I think, also known as Chinese cabbage. I'm not very sure, but this bok choy is a very common vegetable that is present and highly consumed in Singapore. So a lot of these urban farming studies has been in making use of this vegetable for the studies. So biochar is actually produced in the pyrolysis of horticulture, municipal, and agricultural wastes. And this has the characteristics of being lightweight and more durable. So actually in urban farming, so yung characteristics naman ng soil in urban areas are not very suitable for farming, di ba? So and usually you make use of compost to make the soil more suitable for farming. But compost will degrade through time, and so an alternative to that is to make use of biochar. So yung biochar is again more lightweight, more durable. It can last for a longer period of time in maintaining your organic content of the soil for purposes of farming. So we made use of NMR and LCMS for the untargeted metabolomic profiling or untargeted metabolomic analysis of the bok choy samples. So these were extracted using organic solvents and subsequently analyzed. So these are sample spectra in terms of like in NMR. So this is proton NMR spectra, as well as in LCMS. So LCMS, we made use of a high resolution Q-TOF positive and negative ionization. So again, details of these experimental considerations and parameters we will cover in the next session, which will be next week. So after we have obtained the data, we subjected the data into chemometric analysis. So chemometrics is it involves statistical techniques that allows for the simultaneous analysis of different variables. So usually when you work with untargeted metabolomics like these, you generate a lot of data. And in order to make sense of those data, you make use of chemometrics. So we'll again cover more of that in the third session. But for now, this is just to show you that, okay, we have used chemometrics in the data analysis, and one of those chemometric techniques is the use of principal component analysis. So principal component analysis allows you to see patterns or trends that are present in your data. And in this case, you will be able to see clustering of the different replicates for different treatments. So merong walang added na biochar, so 0% biochar. You also have samples with 30% biochar, and you have samples that has 50% biochar. So you can see from here, merong clustering of those that are of the 30% biochar, and those that are grown in the 50% biochar, and meron din for the 0%. So in order to identify ano ba yung mga variables or metabolites that has caused this differentiation into these different groups, you have to look at what is known as the loadings plot. So in the loadings plot, you will be able to see which variables influence the differentiation or the separation of these different groups. So usually, so in expressing or in interpreting the score plots and the loadings plot of principal component analysis, you have to consider same locations will refer to which features are higher in that group of samples. So in this case, are on the right or on my right in the plot will be those that are high in your 30% biochar. So those that are on the upper left quadrant will be those that are high in the 0% biochar, and those that are in the lower left quadrant will be those that are high in your 50% biochar. So the variables here are actually the chemical shift values or the ppm, the chemical shift in ppm values of your NMR spectra. So this region actually covers more of the carbohydrates. So from the spectra, yung mga values na to actually covers the carbohydrate region of the proton NMR spectra. And this is just to show that with application of 30% biochar, there is an increase in these metabolites, mainly in carbohydrates. On the other hand naman, those that are on the lower part are actually corresponding to some amino acids. And again, this shows that certain amino acids, those that are on this region are those that are corresponding to the amino acids. And actually, that means the level of amino acids are higher in your 0% biochar, and the application of biochar results in a decrease in the levels of these amino acids. Now, this is more of the LCMS results. So you look at, first we look at the results of the LCMS analysis in positive ionization mode. So here again, you see some clustering or some differentiation of your samples for 0%, 30%, and 50% biochar. And mainly what you can see is that in terms of the loadings plot, you have an increase in certain flavonoid glucosides for your application of biochar. So for 30% and 50% biochar, you have increased levels of these glucosides, flavonoid glucosides. On the LCMS negative mode naman, again, you see a differentiation of your 0% biochar from your 30 and 50% biochar. Merong mga outliers na kasama dito, but mainly the other replicates are present more on the right quadrant of the score plot. And these are actually influenced by the increase in levels of your some of these flavonoid glucosides, as well as your glucosinolates. So glucosinolates like gluconapine, glucobrasylcanapine, progoitrine, etc. So actually those values here, the number values here correspond to your MZ value, which is your mass to charge value, that can be used for the identification of your compounds based on accurate mass. Again, we are using a high resolution QTOF LCMS system. So you'll be able to use the accurate mass generated for purposes of identification. And we also make use of tandem MSMS. So we make use of information dependent acquisition, MSMS, for purposes of fragmenting your these precursor ions, these molecules of interest, and then from there you'll be able to confirm ano ba yung identity talaga ng mga ions na to. So lastly, this is just to show the effects of biochar treatment in the camphorol biosynthesis. So from the different loadings plot here, you can see that mainly it is the camphorol glucosides that are camphorol and its glucosides that are mainly perturbed in terms of like the pathways that are affected for biochar application. So you can see here which metabolites are higher in the 0%, and subsequently which ones are higher in your 30 and 50%. So this is just to show that with the application of biochar, there is increased glycosylation and acylation of your camphorol metabolite. So actually, in this particular project, we were able to see the complementarity of making use of LCMS and NMR for metabolomic study. You see more of the primary metabolism changes in the NMR side, whereas you see more of the secondary metabolite changes in the LCMS analysis part. So again, this is one application. Another application that I would like to briefly share with you is the use of metabolomics to study the metabolism of tri and butyl phosphate in the earthworm Porionyx excavatus. So tri and butyl phosphate, or TBP, is actually it's a ubiquitous compound. It is commonly used as a component of plasticizer, industrial solvents, and it's also used as an anti-foaming agent. So you can expect it to be present in different environmental matrices, such as in soil, in water, in the air, etc. And so we made use of this earthworm model. Porionyx excavatus is a common earthworm that is found in Asia, in tropical regions like Singapore. And we used untargeted LCMS metabolomics, again, making use of the high-resolution QTOF LCMS for purposes of getting high mass accuracy MZ values, as well as tandem MS information for purposes of later on identifying the different metabolites of these, or of this xenobiotic, the TBP. So this just shows like using untargeted LCMS-MS, we were able to identify certain metabolites of TBP. So this is just showing you the use of MSMS to identify, to confirm the identity of these TBP metabolites. So you can see from the structural elucidation, making use of the fragmentation pattern that is obtained. And we were able to identify some of those metabolites, such as the phosphorylated TBP, hydroxylated TBP, ethanol TBP, and the sulfated ethylene glycol DBP, or dibutyl phosphate. And actually making use of the relative of the quantitative information that you can get from the LCMS, we were able to obtain relative quantitation of the levels of these different metabolites in the earthworm. So we have looked at acute exposures of 1/10 and 1/5 of the LC50 dose for 2 days, and also at the chronic exposure of making use of 10 milligram per kilogram and 50 milligram per kilogram exposure that is incorporated in the soil. So yung acute exposure, we made use of filter paper contact test. On the other hand, yung chronic exposure naman, the TBP was mixed into the soil, and the earthworms lived in that soil environment, contaminated soil environment, for 28 days. So as you can see from here, mainly the metabolites that are higher in levels in both acute and chronic exposures are mainly M2, M3, and M7 and M8. So these corresponds to these metabolic path or metabolic routes, which are mainly the hydroxylation and the phosphate conjugation of your tributyl phosphate. So this just shows, actually, we wanted to emphasize with this research that when you're studying these xenobiotics, you would need to consider the different metabolic products, or the exogenous metabolites of these xenobiotics in the terms of like the risk assessment and safety assessment. Kasi you wouldn't know transformation products of full or more toxic than the main synbiotic compound. So this is again more of an application of metabolomics in environmental research. Now we go to a more details. We make use of I will show you more details in one application of metabolomics in my thesis. Actually, I studied mainly the metabolomics of trauma injury for my thesis, and this has been done in collaboration with the Defense Science Organization, National Laboratories of Singapore. So they are the Defense Science research arm of Singapore, and it has been a privilege working with them. So for this particular presentation, I will present on the metabolomic study of complex combat trauma injury in a porcine model. So porcine model will be a pig model. So trauma injury is one of the leading causes of morbidity and mortality, significant contributions to the society, and this results in significant personal, social, and economic impact kapag nagkaroon ng trauma injury. Now, after a trauma injury, you can envision like this particular illustration here. If you throw a pebble into the pond, you would expect ripples to radiate from the point of impact outwards. And the same goes for trauma injury. So there is a complex network of changes that occurs after a trauma injury. And this can affect organs that are far from the site of injury. So for example, the injury can be just in the head, because of certain physiological or pathophysiological situations or effects. The complications can go up to other parts of the body, such as your kidney, your liver, your heart, etc. So again, these can be normal physiological changes that can proceed uncontrolled, which because they proceed uncontrolled can cause complications, or they can be pathological changes. So pathological changes mainly, for example, the presence of infection, sepsis, and so forth. So one of these normal physiological changes that can be harmful if it proceeds uncontrolled is the inflammatory response. So when we talk about inflammation, di ba, right away, pangit yung notion natin. We think of inflammation as a disease in itself, or as a bad symptom in itself. But actually, inflammation is a response of the body to a particular insult or injury, and it is meant for wound healing and regeneration. So when an injury occurs, there is usually a local response at the site of the injury that is facilitated by cytokines, endothelial cells, and macrophages. And this activates a series of reactions and highly controlled processes in order to restore homeostasis. Now, if the homeostasis is restored, there is resolution and recovery. But if the reactions or the processes proceeds uncontrolled, this can result into systemic inflammatory response syndrome, or SIRS, and subsequently it can affect different organs, and this can eventually lead on to multiple organ dysfunction syndrome, or MODS. So as you can see, the inflammatory response is actually, you can consider it as a double-edged sword. So on one hand, it can be useful. It is actually an integral part of our wound healing response. But on the other hand, if it proceeds uncontrolled, it can lead to a whole slew of different complications. And so, from here, you can see that if an initial injury is not resolved, this can proceed to SIRS and affect remote organs and cause multiple organ failure or dysfunction. So, in combat trauma, there is a higher risk of mortality, of course, and the nature of wounding agents are more deadly kasi nga this is combat trauma settings, and so the weapons are definitely for purposes of like disabling or killing the opponents. You expect multiple wounding in a combat trauma settings of it can be from explosions or from gunshots. It can be trauma from the resources. So hindi available in lahat ng resources that are usually available in a hospital setting. And so there is a need to really study combat trauma injury in order to identify ano ba yung mga pwedeng gawin in terms of like treatment strategies as well as supplementations for combat trauma injuries. And this is also in order to address problems of delayed access to definitive care. Ano ba yung pwede mong i-implement na initial treatments at the combat trauma scenario before they reach the hospital in order to prevent these different secondary complications. So the mechanism of combat trauma injury is usually coming from fragments of explosives. It can also be second to that will be because of like bullets. And in terms of the primary site of injuries, these are usually to soft tissue. So more of your, for example, the abdominal area, your internal organs as well as your extremities. So these are the primary sites of injury. And you can see a prevalence of penetrating trauma in combat injuries. Now, with penetrating trauma, so once, for example, a gunshot or impact of explosives fragment, this can result to serious injury, trauma injury, that can lead to blood loss. So deficiency of oxygen reaching the organs can lead to shock, and this can trigger severe metabolic derangements and a complex inflammatory response. Eventually, this can also lead to the development of SIRS and possibly MODS. And so, for this particular study, we made use of a porcine model, pig model, to study complex combat trauma based on a complex combat trauma model that was established by Cho et al. And why did we use pigs? So actually, pigs are more, in terms of metabolism, physiology, and hemodynamics, pigs are actually similar to humans. So that is why we made use of pigs for this particular model. And this is just to show you a diagram of what has been done. So for the trauma group of pigs, so you have male Yorkshire, I think Yorkshire landrace pigs that are subjected to trauma, and you have a group of pigs that are subjected to the different experimental procedures but not the injury phase of this trauma model. So for this model initially. So of course you have your anesthesia, um, um, treatment, your intubation, instrumentation, and subsequently, before the injury phase for the trauma sample, but blood sampling was obtained for both the sham and the before trauma to serve as their baselines. And then the injury phase consists of penetrating trauma using a bolt gun. And then subsequently, blood is drawn out in order to induce or to simulate blood loss. In this case, they looked at 60% blood loss for this model, and then subsequently subjected the animal to hypothermia. So they reduced reduced the temperature, the core temperature of the animals to around 33 degrees centigrade, and subjected them to 30-minute shock period, which is usually the time period that is considered in terms of like the time by when treatments in order to relieve or to address their injury. Okay, so this is again a combat injury simulation phase. So with this combination of injury, you expect trauma hemorrhagic shock to develop, and afterwards blood is again obtained in order for us to later on be able to compare the metabolic profiles before and after trauma in comparison to your sham, which has been subjected to all the different procedures such as anesthesia, intubation, instrumentation, but except for this injury phase. And in this manner, we'll be able to identify ano ba yung metabolic changes that can be associated with trauma injury. So this is to show the instrumental analysis workflow. So we have plasma samples obtained from the pigs. They were subjected to extraction and put in precipitation using solvent. For the supernatant, this was divided into aliquots for helic or hydrophilic interaction, liquid chromatography, MS analysis, as well as reverse phase LCMS analysis. And a certain portion of the plasma sample was used also for NMR analysis. So this is high resolution proton NMR analysis as well. And subsequently, the data was extracted and analyzed using chemometrics. So again, we will cover more aspects of this instrumental workflow or the experimental workflow for metabolomics in the subsequent session. So this is just to show you an overview of the data processing that has been done. So we have done some data preprocessing and extraction, and subsequently this is subjected to chemometric analysis. You have multivariate analysis such as PCA and OPLSDA. And then because there are so many metabolites that are involved, we further narrow it down using univariate statistical analysis, using Wilcoxon-Mann-Whitney for fold change in order to see fold changes in the metabolic profiles. And we made use of certain databases for the identification of these metabolites. And subsequently, we did metabolic pathway analysis and also correlation network analysis. So this is just to show you an example of the NMR spectra of the plasma metabolic profile of trauma injury. So this is to compare your before sham and after sham, as well as before and after trauma. So in terms of like the visuals, you can mainly see changes in this particular chemical shift at the peaks at this particular chemical shifts for the trauma group of pigs. So using chemometrics, you are able to see a clearer visualization of the changes in terms of your metabolic profile, your after trauma being highly separated from the rest of your sample, from your baseline as well as your sham samples. Now, in terms of the OPLS DA S-line plot, this is actually used in order to identify ano ba yung metabolites that are differentiating your trauma samples from the baseline and sham samples. And here we can see, so this stated here, positive y-axis values is downregulated. So that means these compounds are actually low in your trauma injury samples compared to your sham samples as well as your baseline samples. So it is mainly changes in your proteins, N-acetylglycoproteins, lipoproteins, cholesterol, lipids. So basically, it's mainly lipid changes as well as protein changes that is observed in terms of the changes in the NMR metabolomic profile. So on the other hand, here is an example. Here is the PCA score plots for your LCMS analysis. So again, we made use of a multi-mode LCMS analysis. So we made use of hydrophilic interaction and reverse phase LCMS. Hydrophilic interaction to analyze the polar metabolites, reverse phase LCMS for the less polar metabolites. And we made use of both positive and negative ionization, MS, for the detection. So again, although we have to make use of a 3D plot in order to see the differentiation kasi mas maraming variables yung involved when we're working with LCMS data. So actually, you can generate over 3,000 or 4,000 different variables in some cases. So here you can see also the differentiation of your after trauma samples from their baseline and sham samples. So from here, so this is just to show the different differentiating metabolites in terms of the LCMS analysis, and you can see the diversity of metabolites that has been tentatively identified. You can see changes in your saturated and unsaturated fatty acids, oxo and hydroxy fatty acids, in your purines and pyrimidines, in your amino acids and derivatives, organic acids, as well as in your lipids. So this Venn diagram is just to show that while there are certain compounds that has been identified in one, two or more different ionization modes, there is actually a greater diversity in those that are identified in separate techniques. So this is just to highlight the need to make use of complementary analytical platforms in order to increase the coverage of your metabolome. So this is to show the different pathways that has been affected. So mainly it's the glycerophospholipid, purine, pyrimidine pathways, as well as some of the amino acid pathways. This is just a representation of the different, this is just a combination of the different pathways that has been affected. So I was able to see it or to combine everything in a diagram such as this, and you can see the central role that is played by the methionine metabolism pathway in terms of this response to the trauma injury. We also observed an accumulation of uremic metabolites. So when we talk about uremic syndrome, it is the accumulation of urea and certain metabolites in the blood due to the presence of renal dysfunction or phlegm. Accumulation of these compounds or metabolites is indicative of the presence of possible toxins as well as uremic retention solutes that are accumulating. So this can be indicative of acute kidney injury due to the ischemic shock. And because of this, we see that, okay, there is a possibility that early on, after the shock, trauma hemorrhagic shock, there can already be renal dysfunction that is occurring, or renal dysfunction and other organ dysfunction that is occurring in the pig model. And so we looked further into certain protein markers of like inflammatory response as well as organ failure. So you have cytokines that are related or that are associated with inflammatory response. So these are responsible for the inflammatory response. And you also have like some molecules such as KIM1 or kidney injury molecule 1 and GAL that are more associated with renal injury. You also have those that are more associated with myocardial injury such as CKMB, HFABP, and C-reactive proteins. So from here, you can see those that are colored are those that are higher in terms of the fold changes after trauma injury. So so fold changes is the levels. So levels that are higher than 1 are indicative of higher levels of these markers in the after trauma compared to the before trauma. And these asterisk denotes the significance. So 2 asterisks, this denotes a p-value of less than 0.01. These are Benjamini-Hochberg adjusted p-values. So here you can see that early on there is some indications of the effects in terms of inflammatory response, acute myocardial dysfunction, as well as renal dysfunction in the pig model after injury. So for this one, we just did a correlation analysis trying to identify the associations or relations of the metabolites with these different protein markers. And I just wanted to highlight some of these compounds or metabolites that are highlighted that are significantly correlated with certain for example, here KIM1 and NGAL, which are mainly indicating renal injury. You have 1-methyl uric acid, which is mainly associated with your markers for myocardial injury, such as your HFABP, CKMB, and CRP. And you also have your group of sulfur metabolites that are mainly correlated with your inflammatory response markers, which are your cytokines. And with this, we made use of network analysis. So there is already a paradigm shift. Instead of just looking at pathways, now people are looking at networks. So this is in recognition that these pathways are not existing in itself. It is actually part of this whole network of metabolism. And so there is the interdependence of the pathways, and this is very useful for the integration of omics. Of different omics data. So we made use of correlation network analysis to explain the relationships or interacts in order to gain novel insights into the biological mechanisms, and in order to easier visualize those observed correlations between these two groups of molecules. So in terms of the succinoadenosine metabolites, this is the subnetwork of the correlation network analysis. And in the immediate network or close network of these succinoadenosine metabolite, you can see the KIM1 and the NGAL that is directly and strongly correlated to this succinoadenosine as purine metabolites. And some methylated metabolites. So Actually, if you look at the purine biosynthesis metabolic pathway, succinoadenosine is actually a product that is produced if the formation of AM failure of the conversion of this adenylosuccinate into your ATP. And ATP is a main energy source in terms of the metabolic pathways. And therefore, this elevation of succinoadenosine can be indicative of the dysfunction of this pathway. Unfortunately, actually, the production of GTP is also co-regulated by ATP, and therefore, not only is ATP production affected, but also GTP. And it has been reported previously that GTP depletion of GTP is actually associated with renal tubular injury after ischemia. So this is just to also support the observed possible renal dysfunction in the model. This is just to show the cysteine methionine network. So you can see mainly your cysteine methionine metabolites, as well as your methylated metabolites. And this is very much reflected in the actual cysteine methionine pathway. And this reflects the relevance of the correlation network analysis that we have done. We can see the association or the corresponding, kumbaga, match yung nakikita natin in terms of the network analysis as well as the pathway analysis. This is to show the association of your S-adenosylmethionine and cysteine with the inflammatory markers. So S-adenosylmethionine is actually a metabolite that has been implicated in the modulation of the inflammatory response. And as you can see here, there is a direct relationship of this metabolite with these different inflammatory response markers. This, on the other hand, shows you 1-methyl-uric acid that is correlated or associated with your myocardial injury markers such as CKMB, HFABP, and CRP together in linkage with this 1-methylguanosine. And here we propose some possible mechanisms by which 1-methylguanosine is converted to 1-methyl-uric acid. And this actually implicates 1-methyl-uric acid in possible oxidative stress, apoptosis due to GTP depletion kasi mas mataas yung levels of your guanosine degradation products. And nandyan din yung highly activated inflammatory response as can be observed with increased methylation. So in summary, in this particular application, we have used Metabolomics to obtain a comprehensive profile of trauma hemorrhagic shock using NMR and LCMS. We have identified metabolites that are related to impaired energy metabolism, in particular the depletion of ATP, as well as metabolites that can be related to oxidative stress, the acute phase inflammatory response, and the activation of the inflammatory response. We have also identified the accumulation of uremic retention solutes and toxins that is indicative of possible renal injury. Subsequently, we also used correlation network analysis to gain more insights into the metabolic and inflammatory response to trauma hemorrhagic shock. And from here, we were able to identify potential renal and myocardial injury metabolic markers of this porcine model. And subsequently, we also see the important or the association of your cysteine methionine pathway with the inflammatory response and possible development of organ failure. And so, again, here is the different applications of metabolomics. Again, this is non-exhaustive. Of course, we are very much looking forward to hearing more of your own application, of your own research and how we can apply metabolomics. And Up next, Dr. Fekaling Ashon will be speaking about her own application of metabolomics in rice research, and we can also look forward to the presentations of Dr. Emmanuel Garcia and Dr. Rodolfo Sumayaw in the subsequent sessions on their applications of metabolomics in food and clinical research. I just wanted to acknowledge the OST, PCHRD, and the Balik Scientist Program for this opportunity to be a Balik Scientist. The La Salle University Manila for hosting me as their Balik Scientist. Professor Drexel Comacho, who is instrumental in establishing this Balik Scientist Program for me as well. The LSU SIF staff has always been supportive and is in charge of all the logistics of this webinar series. Dr. Fekaling Ashon, Dr. Mel Garcia, and Dr. Rod Sumayaw for agreeing to be part of this webinar series. And I very much again look forward to hearing about your own applications, and hopefully we can also collaborate in the future. Prof. Sam Lee, my previous advisor and boss, and who has been very instrumental to explore my own research interests, and for that I'm very grateful. Associate Professor Shabir Machala and the DSO Demerit team for who a very instrumental part of my mentors, past and present, that has given me opportunities to explore the different applications of metabolomics. The NUS Environmental Research Institute staff, researchers, and management, past and present, who has been very helpful for me in so many ways in my decade in NUS. So NIRI has been a second home for me. I'm very grateful to them as well. Of course, much gratitude goes to my family and friends, who has been a constant source of inspiration and support. So, and lastly, I would just like to share this slide. This is actually a quotation from the Catechism of the Catholic Church, and actually this is pretty much reflective of my own journey in this research field, and through metabolomics, I was able to see how intricately interwoven everything is, how the regulation, the coordination of these different metabolic networks and the different molecular levels are. And this just puts me in such awe and gratitude to the Lord for giving me this, for leading me into this path, and showing the marvels of His creation in my research work. And for that, again, thank you. That's all. If ever you have any concerns, or if you are looking into applications of metabolomics in your own research areas, please do let me know. How can I be of help? So that's all. Thank you.
Ayun, medyo mind-blown tayo. Thank you, Dr. Karen, for the very informative and solid discussion. Medyo nagsisink in na sa atin yung realization, no, na there's really a lot to learn, but there's also a lot that we can do to level up the research in the country. Plus, na-review pa tayo sa metabolic pathways and instrumentation. So from her talk, we were introduced to the different omic studies and their hierarchies, including a rather in-depth discussion of metabolomics and its applications in biochar for urban agriculture, combat trauma injury. So imagine a metabolomics application relevant to military/defense, and earthworm metabolism, which had also ecotoxicology implications. So in addition, we were also introduced to some data processing techniques like principal component analysis, correlation analysis, network analysis, as well as instrumentation techniques, which we can use for metabolomic studies. Okay, so should you be interested in sending samples for NMR, analytical LC, we have a UV detector, RID detector, and GPC detector, and our soon to be commissioned high-resolution QTOF LCMS for metabolomics and other studies. Just send us a message via email or Facebook. So also, if you have funded studies, we're also open to collaborations. Just hit us up. Ayun, of course, for most of us who are not familiar and weren't able to grasp everything like me, ayun, we can always go back to our FB live recording in the DLSU Central Instrumentation Facility page. So we can use the recording to be better ready for what's in store next week in the Introduction to Metabolomics Part 2. Okay, so anong oras na po? Ayan, mag-11 na. So actually, before we proceed to Dr. Kalingasion's talk, we have a surprise raffle draw. Ayan. So we will be drawing winners from the almost 700 registered participants to the event. So ito yung prizes natin. For the first draw, we will be having a 500 peso globe load card as the prize. This is from the chemistry department. For the second and third draw, we will be raffling out a DKSH or 2 DKSH work from home packages, which we will be sending to your address. Okay, so some raffle mechanics muna. The draw is open to all who registered dun sa webinar natin. Second, there will be 2 winners of the DKSH package and 1 winner of the Globe Load card worth 500 pesos. If you are chosen, please comment, I love Metabolomics doon sa ating doon sa ating chat box. So guys, wag mo na mag-comment ng I love Metabolomics, wala pa tayong draw. Okay, later na po. We will be monitoring both Zoom and FB Live. And number 4, if we don't receive a comment within 1 minute, we will be choosing another winner. So be quick, please. Ayan. So standby tayo. And then lastly, we will be messaging the winners via email at the end of the program. Okay? So are you all ready? And bale, pa-flash muna natin ngayon yung ating raffle sort of software online. And click to spin. So ito na. So for the first winner, mananalo siya ng 500 pesos na globe load care of the chemistry department. Guys, wala pang nadodraw. Ayan, kalma lang tayo. Hi, Miss. Congratulations, Miss Vanessa R. Carbon. Okay, our team will be monitoring for the next minute. Kung mag-comment ka, ayan, sa'yo ang 500. So Miss Vanessa R. Carbon, kindly comment doon sa ating Zoom chat box or doon sa Facebook Live if you're watching. Okay. Meron po ba, Miss Vanessa? And so I don't think meron or I don't think andito siya. Tama ba guys? CIF team. Okay, anyway, let's draw another name. So, are you here, Miss Sol Cristel Magbiro? Sak lang natin, Andito po ba si Miss Sol? And mukhang wala ulit. Sige. Next. Another round. Miss or Mr. Swami Mahadevan Raju, are you here? And mukhang medyo marami-raming hindi po present. So we'll try another one and then if wala pa po dito yung mapipili, we'll skip this round and proceed to the next part of the program. Are you here, Mr. Kenneth Austin Celso? So we have a minute. Here. Ayun. Congratulations, Mr. Austin Celso. We will be sending an email para maklaim mo yung ating 500 peso globe load na prize. Thank you so much. Okay. Sige, sige. Another round for the DKSH webinar package naman. CIF team, kindly note Mr. Austin Celso as the winner for the 500 peso globe load. Next winner for the DKSH package, we have Miss Elvira Bolinguet. Incidentally, she's also a part of Dito Class Tunas Fellowship Program. Are you here, Miss Elvira? Miss Elvi, are you here? Okay, so mukhang wala na siya. I saw her earlier sa list natin. Sige. Siguro lang yung last one. Last tawad. So last draw trial natin. Before we proceed to the next part of the program. Are you here, Miss Maria Lourdes del Mundo? Kindly comment, I love Metabolomics if you're here, Miss Maria Lourdes del Mundo. Okay, so if you're not here, we'll have more prizes for the next 2 sessions of the program, okay? So we're ending the raffle session here and we'll proceed to the next part of the program, okay? I'll now be introducing the next speaker, Dr. Maria Fe Kalingacion. So we are in no shortage of awesome research scientists as we welcome another accomplished researcher in the field of metabolomics. Our guest is an associate professor of the DLSU Chemistry Department. She earned her Bachelor of Science degree in Chemistry and Master of Science degree in Agricultural Chemistry from the University of the Philippines, Los Baños. She then went to Europe to earn her PhD in chemistry from the Wageningen University and Research Center in the Netherlands. Soon after, she took her postdoctoral fellowship at the University of Queensland School of Agriculture and Food Sciences in Australia. At DLSU, she primarily handles the graduate course on Advanced Analytical Chemistry, which focuses on instrumental methods of analysis and chemometrics. An accomplished researcher in the field of rice research, including metabolomics and genomics, she has over 20 publications on the said topic with over 800 citations. So that's no small feat for a researcher. Without further ado, let us all welcome Dr. Maria Fe Kalingacion.
Good morning, everyone. Let me share with you how we have used metabolomics to understand rice grain quality. Rice is life to billions of people, and the population of rice-consuming countries continues to increase every year. By the way, this is not my child, but my daughter loves rice the same way that this child enjoys his rice meal. The rice production every year also increases. In the recent report published by Food and Agricultural Organization, the total production of rice paddy from 2018 to 2019 is around 759 million tons. 89.7% of it are produced from Asian countries, while 4.9% and 4.8% are from America and Africa, respectively. Clearly in this map, it shows that majority of the Asian countries produce more than 3.4 million tons of rice every year. And 2.9 kilocalories or 2,868 kilocalories of the energy that we need are from the cereals. 20% of it are from rice. There's a study shows that although the population in rice consuming countries increases, some of the consumers from developed countries started to diversify their meals and an observed decrease or decline in the rice consumption every year. However, for the Philippines and other developing countries, the increase in rice consumption increases as well every year, okay, as our population increases. This is a good illustration of how from 1980 to 2010, how much rice we have consumed and the increased consumption every year from 92 kilogram to 119 kilogram every year. This is, by the way, for every Filipino's average yearly consumption. 119 kilogram is equivalent to 5 cups of rice. I'm not sure about you, but I'm trying my best to lessen the number of cups I eat every day to help the Philippine agriculture. Rice is also considered scarce. There is a challenge that we have to produce rice under fewer land and less labor. It's because mainly of industrialization and less people are engaging in agricultural activities. This is a good illustration where rice, wheat, and maize rice farms reduced significantly from the 1960s to the year 2000, and significant decrease by half of the hectares of land dedicated for agricultural production. However, there is a good news. The Department of Agriculture made its press release in May 2020 that it says that we have enough rice supply adequate for the whole year, and I hope it will last until the 2021. And the Department of Agriculture is maintaining its goal of keeping a 2.5% growth every year, increasing as well the 2022 budget. Okay. I guess the rice is scarce is not real for the Philippines, so I should write here, rice is enough at least in our country. Growing rice is a challenge. Cultivation of rice under suboptimal conditions are the challenges that the farmers as well as the rice breeding programs are currently facing. To produce 1 kilogram of rice, it says that we have to utilize 10,000 liters of water. That's a lot. But recent advances in breeding programs like marker assisted or molecular breeding were able to develop different types of rice that can adapt to different conditions, such as rice varieties that can tolerate submergence or flooding, mainly because of the presence of a quantitative trait loci or a gene responsible for flooding tolerance or submergence tolerance, as well as rice varieties that can withstand very minimal water, supply of water, mainly because of the drought genes. Some rice varieties are also being developed to cater for drastic conditions like high salinity and pest infestations. So if you look at it, yes, there is a challenge, but science is doing its part to continue to address to this moment, this big task. For rice chemists like me, it is our goal to provide information on how we can produce good quality rice, not only of amount, but also of quality. That is our goal. Majority of the rice breeding programs have rice grain quality laboratories that do the routine analysis for them. Among the properties that they analyze for breeding programs are the physical properties and the cooking properties. Physical properties are mainly focused on grain length, shape, and chalk. These are the characteristics that consumers um, easily see at the market. So definitely the physical properties command a higher value when you are dealing with prices in the rice market. The length of a grain refers to the dimension of the grain itself. Surprisingly, for us in the country, we are more often introduced to rice varieties that are long and slender. But actually, rice grains comes in different forms, from short, medium, long, and very long as its length, and bold, medium, and slender for its shape. By the way, shape is defined as the ratio of the length and the width of the grains, where we have bold, medium, and slender. Why are these shapes important, or why are these dimensions important? Mainly because of cultural preferences. Majority of the Southeast Asian countries prefer the long and slenders like this basmati rice, while for Eastern countries like Japan, they would prefer the short and bold type of rice. If you notice the sushi that we eat in Japanese restaurants, they are the cute grains that we see because of those short and bold shape. Chalk, on the other hand, is the opaque um, appearance that you see on the grains and the sperm. The chalkiness affects the overall look as well as the um, tendency for the grains to break when they are milled or polished. Of course, if you see broken grains, it commands lower value than those grains that are whole or intact. That is why when you see um, grains or a bag of rice that are full of broken grains, it is usually of cheaper price compared to those that are of whole grains. And chalk is playing an important role in that. Aside from the physical properties, we also looking at the cooking properties of rice, such as the amylose content, gelatinization temperature, and gel, gel consistency. So amylose is a linear polymer composed mainly of glucose units linked primarily by alpha 1,6 linkages. If you say that there is no amylose or waxy, these are the waxy rices, or we popularly known in the Philippines as the malagkit rice. So waxy rices are also popular in Southeast Asian countries like Laos. Low, intermediate, and high are those rice varieties that have amylose content ranging from 0 to 20 percent. And very high means it has more of the amylose than the amylopectin. What does amylose mean? It means that it's the tendency of your, aside from the stickiness that we observe in our malagkit, it's also the tendency of the rice to retrograde after cooking. Okay, retrograde means namamahaw or tumitigas after cooking. Gel temperature, on the other hand, is the temperature at which the grain starts to cook. It is where the crystalline structure of your grain starts to melt and your rice starts to cook. Okay, so we have gel temperature ranges from low, intermediate, and high. Usually, this is from 55 degrees to 85 degrees Celsius. Okay. Did you know that lowering the gel temperature just by 1 degree Celsius, or just 1 minute, I mean, so assuming that there are millions of household cooking rice and reducing their cooking time by just 1 minute, it would mean 25,000 years of energy less every year that is conserved. Aroma, on the other hand, is one of my favorite topics. I guess you'll notice that the majority of the topics that I will be discussing in the coming slides will be dedicated to the aroma of rice. Aroma in rice grains are usually just done either to classify the rice as fragrant or non-fragrant. Usually you do it, you put 1 gram of the rice grain, soak it with potassium hydroxide for an hour, and then you sniff it. But of course, it takes some time for a trainer to identify whether the rice variety is fragrant or not. And of course, just like this, sniffing also would result to oversaturation in the detection of the fragrance of rice. Recently, routine analyses are now being done to monitor the 2-acetyl-1-pyrroline, or popularly known as 2AP by gas chromatography. 2AP is what 2AP is the compound responsible for the flower, the jasmine, and the pandan that we usually smell when the rice is cooked. Actually, for aromatic rice, if you visit the field, even if the rice is not cooked and it's still planted in the field, you can still smell this pandan flavor. That is how strong 2AP is. So these are the common quality traits that we monitor when we ensure that the rice varieties that are being developed in breeding programs are of good quality. With all of these traits, it's still surprising that the actual rice market is not represented properly. Okay? What do I mean by this? For the past with For the past few years that I've been working with rice, I've been part of the International Network for Quality Rice. These are chemists, manufacturers of machines, and rice breeders from 31 countries, all working towards producing quality rice. And from this survey, we have identified 3 famous rice varieties in each country, in each of the member countries, and summarized the common traits, such as the physical and cooking properties. Consumers within a region or within a country, if you'll notice here, have quite a diverse rice preference. Amazingly, for India, you'll see here the diversity of the shape, the length and the shape of the rice grains that they prefer. The bottom or the southern part would prefer the short and the um, bold type, while the northern part would prefer the extra long and slender type. Similarly, in Southeast Asian countries, where Philippines is part of, the amylose content is also quite diverse. Waxy rice or zero amylose is preferred, as I mentioned from the previous slide, is preferred in Laos, while low to intermediates are preferred in Vietnam and Cambodia, while Philippines as well as Thailand would prefer intermediate to high amylose content. Okay? If you summarize all of these traits, combine them in one design, combining the 4 properties of length, 3 properties of shape, 4 amylose classifications, 3 gel temp, and 2 aroma, will come out with 18 trait combinations. And these are the countries and their rice varieties belong to. Amazingly, with 18 trait combinations, we thought it is enough to define or describe a particular rice variety. But no, we were surprised that for the Philippines and Brazil, particularly IR64, and Brazil's BRS Primavera. They both belong to long grain, slender, intermediate amylose content, intermediate gel temperature. It is considered non-aromatic, but people would know the major differences between an IR64 and a BRS Primavera. With this, it is not surprising that we need additional tools to define rice grain quality. And this is where metabolomics comes in. As has been defined, metabolomics is a tool for comprehensively profiling small molecules. I love this illustration because it says the hierarchy from the DNA, from the genetics, up to the metabolites. And what is good about this illustration is the metabolome is what is dictating the phenotype or the characteristics or the traits that we are seeing, particularly in rice quality. So with this, let me share with you some of the applications that we have done using metabolomics to understand the volatile compounds using a single platform. Several properties such as elements, volatile compounds, polar and semipolar metabolites, as well as primary metabolites using multiple platforms. And finally, combine genomics and metabolomics as a multi-omics approach and be able to identify the volatile compounds as well as QTL or the quantitative trait loci mapping that will be valuable in molecular breeding programs. Let's do first the single approach in volatile analysis. In any breeding program, you always start with the right parents. I guess in any partnership or relationship or any endeavor, you always look for your complement. In this population or in this study, IR64 and APO or ERI-132 or also known as NSIC RC9 for commercial code are being developed at this time, some time ago, to capture the good traits of each of the parents. IR64 is one of the elite rice varieties in breeding programs. It's because of its good grain quality traits. However, like any other elite rice varieties that were introduced, by the way, IR64 was only introduced in 1985, they are quite susceptible to pest infestations, to drastic environmental conditions. That is why the yield are not always favorable. However, IRRI-132 or ERI-132, or better known as APO, let's call it APO, are very resistant rice varieties. They can withstand low soil water or moisture, like in irrigation. So we can safely say that it's considered as a drought tolerant rice based on this graph, where the yield of IR64 irrigated as well as the IR64 drought have significant difference. That's why we can consider IR64 as a susceptible, drought susceptible rice variety. Apo, on the other hand, with Apo irrigated and Apo drought, the yields have differences, but these are not significant, which means that Apo can withstand drought conditions or can resist low water supply. If we look at their grain quality traits, IR64 as well as APO have similar gel consistency, gelatinization temperature, as well as the amylose content. So if you just look at the gel quality, the grain quality traits, you won't be able to distinguish IR64 and APO. However, when you do sensory analysis, as we have done here, we have conducted and trained panelists to differentiate rice grains from apple and IR64 grown in irrigated and drought conditions using several descriptors like taste descriptors like the sweet taste and the sour or silage taste, mouthfeel like the astringent and water-like metallic, and the mouth and other descriptors like aroma like corn, sweet aromatic, sour animal, and hay-like and musty. So how do we come out with this is because previous analysis by Champagne and her group already conducted a sensory analysis of Apple and IR64. So to lessen the uh, number of descriptors that the panelists will be considering, we only focus on these eight sensory descriptors. And true enough, when we see, when we compare the IR64 irrigated and drought grains, there are significant differences with the sensory results, you know, where majority of the astringent, water-like, sour, and sour are missing in irrigated IR64. However, for apple, whether it's in irrigated or drought conditions, you won't see much differences between the grains. By the way, the plus signs indicate the intensity of the descriptors as the panelists are describing them. When we do the metabolomics of the volatile compounds, we came out with, we were able to identify around 187 headspace volatile compounds. This is the principal component analysis of 12 biological replicates of APO and IR64. So the blue ones are the IR64 irrigated, the green one is IR64 grown in dry condition, while the red and the gray ones are the apple grown in irrigated and dry conditions. You will notice that IR64 grown in the irrigated significantly have a different volatile profile than those of the IR64 grains that were grown in dry conditions. But you won't see much differences between the volatile compounds that were emitted by the headspace of the rice grains grown both in irrigated and dry conditions of the apple rice variety. This clearly shows that the metabolite profiles that we identify can approximate or agrees with the results of our sensory analysis, which is quite interesting because conducting sensory analysis is quite tedious and it takes some time to be able to train panels expert for a particular sample and particular study. Another example of using metabolomics is using multiple platforms. Um, with the collaboration, we were fortunate enough to be part of the Metaphor project, Metabolomics, with partnership with different research institutions in Europe. We are able to identify different properties of rice grains, such as multi-elements, using ICP-MS from University of Copenhagen in Denmark, volatile profiles of our rice grains based on the GCMS solid phase microextraction done by Wageningen University in the Netherlands, um, polar primary metabolites by GC, time of flight mass spectrometry done by Max Planck Institute in Germany, semi-polar and polar metabolites as done by nuclear magnetic resonance in the laboratories in Rothamsted, and direct infusion mass spectrometry both in the negative and positive mode in also in Rothamsted. Okay, so with this, um, different platforms, we were able to see the effect of different parameters like storage. What happens when a group of jasmine rice varieties are stored, um, and how are their volatile, since jasmine rice are very famous aromatic rice. Um, what is the effect of storage with its overall fragrance? This is the composition of all of this um, um, measurements, and came out with a principal component analysis that looks like this. Okay, for freshly stored or freshly harvested jasmines like RD6, KDML 105, or Kaudak Mali 105. This is the elite jasmine rice variety. RD33, as well as the Patam Tani 1, which is also the second best jasmine rice in Thailand. Clearly, their profiles are clustered together, which means they share almost similar characteristics, whether it's the volatiles, polar or semi-polar metabolites, as well as their elements. And as you increase the months of storage from 6 months to 12 months, you'll see a clear differentiation between the stored grains as well as the harvested jasmine rice varieties. With this, we can say that metabolite profiles indeed can identify whether rice is stored or freshly harvested. And we can use this information to um, see, determine the proper post-harvest treatments to ensure that aromatic rice like jasmine are kept in its finest quality. If we are to compare now two very famous rice varieties like jasmine and basmati. By the way, this picture was taken during one of the conference that were majority attended by members of the INQR, or the International Network for Quality Rice, as well as the Metaphor, who analyzed the rice varieties. There was a raise of hand or a survey of who prefers jasmine. So this is our good friend from Thailand, who proudly raised her hand to show her love for jasmine rice, while this is our another friend from Iran that would prefer basmati. So she's waiting her turn to raise her hand for basmati. By the way, I am the lady at the back also raising my hand in favor of jasmine over basmati. So if we compare these rice varieties based on their location grown or their origin, you'll see a significant um, clustering between the jasmine and the rice and the basmati. Here, you'll see the RD33, KDML 106, 105, RD6, as well as the PTT1, all clustering based on their volatile profile. Similarly, for their polar primary metabolites, as well as their semi-polar and polar metabolites, you'll see a cluster of jasmine in one side and another big cluster of the basmati rice varieties on the other side. By the way, the rice varieties of the basmati rice varieties are from members in from Iran and Pakistan, who gave us the different basmatis like basmati 370 um, and Supa basmati. And if you notice, there's a indication here that these are the fresh basmati. We clearly indicated that we want to compare all the fresh samples. No, it was difficult at first to find fresh rice, basmati rice, because the basmati is commonly stored for a year before they are sold in the market. So if we look at the um, basmati that are available in the market, they are usually characterized by off flavors or off scent. No? But they are stored, as I found out from our friends from Iran and Pakistan, as well as in India, that these rice varieties are stored because of the development of a better texture that are needed for their local cuisine. Okay, so they are not actually after the aroma, but rather more on the texture. Okay. So with this, we can say that yes, the metabolite profiles can actually differentiate rice varieties depending on their origin or their location. What is the practical application of this is we can identify whether rice basmati that is claiming to be from Iran can be identified based on selective aromatic compounds that are unique from basmati that are purely from Iran. Okay, so contaminations or adulterations can be monitored. This is one good application of this type of information. Similarly, IR60, no, not IR64, but rather the KDML 105 or the jasmine that is commonly sold in the market are also adulterated and contaminated with other non-aromatic um, rice. So it will be a very useful information if we'll be able to identify um, markers, volatile markers for this particular type of rice. Okay, so now we have shown that single and multi-platforms can differentiate rice varieties, whether it's their tolerance to drastic conditions or their locations or grow or yeah or their origin. Okay. Another approach that we also want to look at is the multi-omics, no? Combining genomics as well as metabolomics. The genetic basis of rice aroma based on 2AP is already known. We already know that the betaine aldehyde dehydrogenase gene is responsible for the production of 2AP. That is why after the discovery of this gene, now there are three ways of identifying rice varieties, whether they are fragrant or not, in a rice breeding program. First is the sniffing, the second is the GC, and the third is through this molecular mapping approach. Okay, so how does it work? So we have in rice, we have 12 chromosomes. On chromosome 8, in its exon 7, there is a deletion of um, information. This deletion results to a non-functional gene. So if the gene is functional, that means it's complete and there's no deletion, the betaine aldehyde dehydrogenase will produce GABA or gamma aminobutyric acid that is used when the rice plant is under stress. Okay? But if there is a deletion, especially on this region, there is a malfunction or non-performance of this gene, resulting to an aromatic rice or the production of 2AP. This deletion is currently being developed as a screening tool and expand the capacity for early detection of fragrant rice varieties. Because with this method, you do not need to mature the plants, harvest the grains, and sniff or analyze it through the GC. With the DNA extracts, you can identify these alleles and be able to classify whether your rice at an early stage is fragrant or not. So from these publications, um, there are around 10 alleles that were identified, whether it's a deletion, insertion, or a change of SNP, single nucleotide polymorphism of G to T or C to T. So these alleles are now being used routinely to screen for early stages of rice varieties. It's really cool. There is a big advantage for this. However, this is like any other rapid test. You won't be able to quantify the level of 2AP that the rice variety is producing. Unlike for gas chromatography, which is I am biased for, can quantify the amount of 2AP in per every gram of grains produced. Okay, so we have done drought conditions in the Philippines. We have also analyzed using multi-platform metabolomics using rice varieties from Southeast Asia and South Asia, so from mainly Thailand, Iran, and Pakistan. Now we're moving towards South Pacific, mainly in Australia, where there is a breeding program to develop fragrant rice varieties using elite lines from Thailand, Laos, Cambodia, and use non-fragrant rice varieties that are of good quality, like IR64, Dungara. Fragrant rices also from Australia, like Kayima and Pandanwangi from Indonesia. So this is part of a breeding program, and we want, because of its unique um, collection of all of these combinations of parent rice varieties, we can actually understand better if there is a um, other genes that are responsible for 2AP. Okay. Analysis of these compounds, analysis of these rice varieties and their progenies from the different populations resulted to this principal component analysis, where we have several compounds. Majority of the fragrant rice varieties are on top of the PCA, while at the bottom of the PCA are the non-fragrant lines. Interestingly, this is actually a biplot where you can see the rice varieties as well as the compounds or the volatile compounds that were analyzed from the headspace of the rice grains. So these are the triangles that you'll see in this PCA. What is interesting about this is this set of compounds. If you notice here, the triangle coded as 1 is actually 2AP. And it's not alone in the PCA. Clustered with 4 other nitrogen-containing compounds. 3 that we were able to identify, like pyrrole and 2-acetylpyrrole, but 2 that were needs further identification. So what do we do with these 2 unknowns is analyze them using proton NMR. We synthesize 2AP, and surprisingly, every time we synthesize 2AP, there is another peak that comes out. And this could be the analyte 5, which is 6-methyl-5-oxo-2345 tetrahydropyridine, or probably it's better to just call it 6-M50-TP. Okay? And the analyte 4, we submitted it for further analysis using a 4D GC high resolution time of flight mass spectrometry, or a GC by GC HRT 4D. And from here, we were able to narrow down the identification of analyte 4 as 1-pyrroline. So all of these are nitrogen-containing compounds. And if we look at the genomics using genome-wide association analysis, surprisingly, these 2AP and the rest of these 5 and these 4 containing compounds lie in the same region or co-localized in a single QTL. This is marked with this yellow triangle here. And if you trace it with all of the populations that we have analyzed, let's trace this one. So all of the populations with 67, 71, 21, 36, and 25 progenies or lines are all positive with 2AP. While those that have negative 2AP, this QTL does not exist. which is coded in green color here. So exciting. So metabolomics have proven that these five nitrogen-containing compounds are similar, and also their region in the chromosome are also similar, which might lead us to would they share the same biosynthetic pathway. So this is the published biosynthetic pathway of 2AP in 2008, wherein 2AP is produced from proline, ornithine, and then putrescine when it is converted to aminobutyraldehyde, and the BADH2 gene is functional, it will prefer the production of gamma aminobutyric acid, as I mentioned from the previous slide. However, if there's a deletion in the exon 7 in chromosome 8, the gene is non-functional, it's not working, so aminobutyraldehyde will form a cyclic compound, which is the delta pyrroline, and later on acetylate to form the 2AP that we are looking for. So with this mechanism, will there be another biosynthetic pathway or a modification of this biosynthetic pathway to understand how these five nitrogen-containing compounds are related to one another? And thus we propose the following mechanisms. We also believe that it's always from putrescine and aminobutanol. Okay, and we know that when it's there's a functional BADH2 gene, it will produce the GABA, but when it's not, it will produce the 2AP. When it forms a very unstable 6-amino 2-hexane dione, it will not stay in this compound, but rather it will immediately form a cyclic like 2AP, 2-acetyl-1-pyrroline, or the other compound like the 6-M5-OTP. Similarly, from 4-aminobutanol, you can also form 1-pyrroline, or you can also form it from pyrrole. Okay. And from 2AP, we can also produce 2-acetylpyrrole upon oxidation. So using metabolomics and genomics, we were able to propose another or modify a biosynthetic pathway for the production of 2AP and the other volatile compounds. And lastly, we know that rice aroma is not just about 2AP. We know that when we smell rice, it's not only the pandan smell that we can detect. So what we've done is move to a nearby country called Cambodia and use their world's finest rice variety called Pakarum 2 and couple it with an agronomically good rice like Tamar Creme or TMK and see how the metabolite profiles of or the profiles of their volatile compounds look like and see their genetic if there's also some genetic information about it. So again, similar to what we have done from the previous studies, we have analyzed it using the headspace of the rice grains and use a two-dimensional gas chromatography to efficiently separate the volatile compounds. Surprisingly, from this analysis, we have seen that there are significantly a lot of volatile compounds that are present in the rice grains, not only 2AP, although 2AP is the leading rice volatile compounds here with pandan described as pandan or the sweet flowered smell. Others are also present like pentanol, which is considered to be with an other descriptor of green or grassy, heptane, which is quite sweet, and others would have some wine or nutty scent. If you also notice here, with the next to the other descriptors are the other threshold. Other thresholds in terms of the fragrance are quite important. Other threshold indicates the concentration that is um, needed for our humans to be able to detect. What is good about 2AP is it has a very low odor threshold of zero point 0.1 ppb, which means that even at a very small concentration, you'll be able to detect 2AP in rice grains. Unlike other compounds like this one that has a 2,500 odor threshold, that means it has to be present at that amount to be able to be sensed or be detected by our human sense. Okay, so using that population, uh, you're able to see a PCA that clearly divides the rice varieties according to the presence or absence of 2AP. And from here, we were able to see different volatile compounds like alcohols, aldehydes, carboxylic acids, esters, furans, hydrocarbons, ketones, and the nitrogen-containing compounds. If we look at the cytoscape or a method of determining which of these compounds are related, you'll also see that 2AP as well as the other nitrogen-containing compounds like pyrrole, pyrroline, and the 6M5-OTP are also correlated. If we look at the um, SNP genotyping using Illumina Infinium, using a 6,000 SNP big sheet, and TAAS and QGene for QTL analysis, we are able to construct the following Manhattan plots where majority of our aldehydes and with an LOT of more than 3, which is quite good, are detected here. So here are pentanol, hexanol, heptanol, octanol, nonanol, and these are quite important in terms of their contributions to the overall aroma. And these are quite useful if we will consider a particular rice variety to be developed and that would have high amounts of this aldehyde. So in summary, single and multiple platform metabolomics can reveal unique metabolite profiles of rice, whether it's based on their origin or location or they're grown in different environmental conditions. And we have proven that metabolomics combined with genomics would be a powerful tool to identify metabolite QTLs that are important in marker-assisted breeding programs, and also, it helps us formulate new hypotheses for aroma production or biosynthesis. And we have proven that using multi-omics approach, it will uh, surely pave way to better understand rice grain quality. With this, I would like to thank the following: the International Rice Research Institute, Plant Research International, and the Plant Physiology Department of Wageningen University, the School for Agriculture and Food Science of University in Queensland, and Monsanto Bichel Borlag for my PhD, and Agents of Change Industry Transformation Training Center of the Australian Government, Australian Research Council for my postdoc fellowship. Again, thank you for listening.
Yun, salamat po, Doc Fe, for the wonderful presentation. And salamat din sa genetics review. So actually, I was able to sit in one of her classes, and her lecture on PCA was really helpful. So then, ang ginamit niya was, as an example, was rice metabolomics also. So recap, from her talk, we were able to learn a couple of new information about our beloved rice grain. So which includes characteristics on length, shape, gel temperature, amylose content, aroma, and global preference for the introduction. You also learned the role of metabolomics in rice research. In applications such as single, so volatile compounds, GCMS, and multi-platform approach, volatiles, metals using ICP-MS, GC-TOF-MS, NMR, and direct infusion MS, as well as the multi-omics approach. You also learned about the genetic basis of aroma, which was processed via proton NMR, 4D GC, wow, headspace GCMS, PCA, and genome-wide association analysis. The studies provided insight on chromosomal and biosynthetic pathway implications, as well as the concept of odor threshold, which was introduced earlier. So regarding the multi-omics approach, I actually saw a paper before na it presented an omics studies tapos NMR, and then the objective was to determine the country's country of origin of coffee. So that would be interesting. So if you're into coffee, we'll have more of that metabolomics application next week. Okay, so at this point in time, we would like to begin reading and responding to some of the questions that you have posted. So at the moment, you only have one for Zoom and a couple from the Google form that we made you register or sign up for. So may I invite Doc Karen and Doc Fe to open their cameras for the Q&A portion. Hi, Doc Karen, Doc Fe. Are you here with us?
Hello?
And hi, Doc Fe. Doc
Hi,
Karen.
Chris. Can you hear me?
Yes po, we can hear you.
Okay.
And,
Hello po.
and Laguna represent. So Doc Fe is from Los Baños. Doc Karen is from Santa Rosa, and I am currently in Binyan. And so kaway-kaway sa mga taga Laguna dyan.
Laguna.
Okay? Hello, hello. At sa lahat po ng nanonood sa buong Pilipinas at buong mundo, syempre may mga foreigner ano rin tayo, guests here. So for the Q&A, we will try as much as we can to answer all the questions and address your concerns. If in case we run out of time, we will respond to you separately via email. So let's, without further ado, let's proceed to the first question. This is from Zoom. Hi, may I know the softwares or websites normally used for the untargeted metabolomics in which you can incorporate the bioactivities of the samples? The question is from Mr. or Miss Sydney.
Okay, Doc Fe. So, shall I answer first? Okay, so, Sydney, if you're dealing with bioactivities, I'm thinking, I'm assuming that this will be more of quantitative values, diba? So, actually, you can explore Metabo Analyst, which is a web-based, um, server-based software. It has some capabilities for processing these types of data, wherein you have metabolomics data together with some metadata. So I think they have functionalities for that. Another option that I think you might want to explore is the use of multivariate regression. So merong mga tinatawag na OPLSDA, PLSDA regression that you can use to correlate yung data mo with regards to your bioactivities versus the metabolome. You can also consider correlation analysis in some cases that can be applicable also in some grouping your quantitative bioactivity values into certain quantiles or percentiles, and then making use of that for your PCA analysis. So these are some of the ways that I think you can explore for data processing. And I hope you can join us for the third session. So we'll try to cover more of these in the chemometric session of this webinar series.
In addition, pwede mo rin gamitin ng R kung you're into you're more familiar with programming languages. There are several scripts in R that you can also use. Kaya lang, personally, I'm not hindi ko naiintindihan yung mga computer languages. So I'm more into the web-based, like Metabo Analyst. There are also some that are with licenses or subscriptions that we can also um, tap, like Simca. Also, madami actually. If you want, we can send you a list of these softwares and just let Chris or the other staff of CIF, and we are happy to send you a comprehensive list of the softwares that are available for you.
Yes. So actually, yung Metabo analysis is R-based. So you can actually make use of the Metabo Analyst R package if you're familiar with R. And yun nga, yung sinabi ni Doc Fe, yung SIMCA, it has those multivariate regression functions like PLSDA and OPLSDA regression. So yun.
Thank you for your answers, Doc Karen and Doc Fe. So let's proceed to the next question. This is from Christian Condez. How can metabolomics be used to correlate assays and fractions for bioactive compounds? Again, how can metabolomics be used to correlate assays and fractions for bioactive compounds?
Okay. So, Christian, I think, are you referring to how can we make use of metabolomics analysis in conjunction with the results that we get from bioactive bioassays? Is that what you're looking into? Okay. So, actually, di ba, Christian, when you make use of bioassays to determine the activity of these different functions. In some cases, you're still not sure of ano ba yung components of that fraction. And so in metabolomics, in the use of untargeted metabolomics, for example, making use of, for example, NMR or LCMS or GCMS, you will be able to identify the contents of those fractions. Another application, I think I have this one project wherein we looked at the um, antioxidant activity of this herbal medicine. We made use of Spearman correlation and some PCA. And from there, you can actually see wherein yung, for example, in that particular in one area and those that are of lower antioxidant activity in another area, and these are actually correlated to the presence of certain metabolites. And this is just an example of how you can use metabolomics for determination of the bioactivity and what are the metabolites that are of interest in those bioactive fractions.
In addition to that, you can also use the process called de-replication. So when you have the metabolomics as well as the bioassay results, there are de-replication databases that you can match and will help you identify your active components. In that way, medyo mayroon ka ng assistance on what compounds are you looking at instead na you're doing it alone and at loss. So there are several approaches of or types of de-replications that you can actually be used as a guide when you're doing bioassay and metabolomics at the same time.
All right, thank you, Doc Fe and Doc Karen. So again, that's the replication. So for the next question, we have what are the best instruments to be used if we are doing a comprehensive metabolic profiling? This is from Justin Rotairo. Again, what are
Okay.
the best instruments? Okay, sige po.
So medyo meron kaming kanya-kanyang bias ni Ma'am Pe. So si Ma'am Pe is actually more of GCMS, di ba, si Doc Pe. I'm more of an LCMS person. But actually, we'll cover this in the next session of the metabolomic session, yung sa next Saturday. We'll talk more about the different, they have their own disadvantages and advantages. There are metabolites that can be covered using GCMS, but there are also metabolites that are mainly more suitable using LCMS. We'll cover kung ano ba yung mga criteria, kung ano ba yung suitable for GCMS and LCMS in the next session. So we'll talk more about that sa susunod na session.
And aside from that, hindi lang LC and GC yung pinag-uusapan ng
Mm.
chromatography here.
Yes, yes.
We are also looking at when you're attending conferences, actually, ang main division is not an LC and a GC, but rather an MS, mass spectrometry, and NMR, nuclear magnetic resonance spectrometry. So, yun yung major divisions ng mga tao. So,
NMR.
in terms of that, dun kami nagkakasundo ni Doc Karen, pareho kaming mass spec person. Well, she's doing NMR, but I'm solely focusing on mass spec. So meron ding advantage, disadvantages of both platforms.
Oh, actually, yun. So meron yung mga more of the European proponents of Metabonomics are actually more of NMR-based people. More of those yung nasa mga US-based are more of the mass spec-based. But as Doc Fe has mentioned, ayun, nandun yung people who are more of NMR Metabolomics people. There are those that are more of mass spec-based. And we'll discuss yung disadvantages and advantages of using both or either dun sa susunod na session. So I hope you'll attend that next session, Justine.
Alright, thank you, Doc Fe and Doc Karen. So, ayun, again, dalawang klase, NMR and mass spec. And luckily, if you're from DLSU, especially if you're a grad student, you have access to our NMR, our GCMS with Headspace, and also our QTOF LCMS, which will soon be commissioned. So, in addition, actually, everyone is invited to submit samples din naman. Okay, for a fee, of course. So, everyone na interested can use the equipment. Okay, for the next question, this is from Tecton. Can we use metabolomics to mix non-seasonal crops into all-season crops? Again, can we use metabolomics to make non-seasonal crops into all-season crops? Sige po, Ate V.
Siguro start tayo kay Doc Fe. More of yung sa rice research eh.
Medyo mahirap yung tanong kasi 'di ba, yung season it's because of the weather condition kaya nagfa-flower yung plants. So maraming factors. Actually, marami ng efforts ang ginagawa in terms of rice breeding programs na paano ma-improve ang rice kahit hindi siya sa ganitong klaseng conditions. So, pwede din actually using metabolomics. I couldn't answer it directly, but I think it's possible when you look at metabolomics as well as the genetics. So it's always a partnership. As Doc Karen has mentioned, it's more of a systems biology approach na magkakasama yung genomics, transcriptomics, proteomics, as well as metabolomics to completely understand yung biology of fruit flowering or flowering ng tree to produce fruits. Ganon.
To add to that, there are certain applications right now that are looking into metabolic engineering. So the use of like looking at metabolic pathways and how can these be manipulated to produce certain improvements. But I think so far, in terms of like seasonalities, so far I haven't seen any applications on that. And as Doc Fe has mentioned, it has to be looking at your genes and most likely the whole omics. From your genes to your transcriptome to your proteome up to the metabolome level. So that will be a very big effort. So hopefully you'll be interested to pursue that. Yun.
All right, thank you, Doc Fe and Doc Karen. So we have another question, but this may not be that related to the topic. Where can we find published rates for CIF services? So, Mr. Angelo, sir, we'll send you a message to address this concern. For the next question, this is from Mr. Zorel Alegre. What topics for a metabolomics research for a food science major thesis are possible? And what equipment would we need?
Okay.
Send an email at mariafederation@lsu.edu.ph and I'm happy to collaborate with you. medyo marami tayong pwedeng gawin with
Ayun.
metabolomics in food science. And ang daming equipment sa central instrumentation facility that we can tap for any food science related projects.
Ayun. And ayun, as Doc Fe has mentioned, you can let us know how we can be of help to your thesis. Again, I'm a balik scientist. I'm not really a faculty right now, but if in any case I can be of help to your research, pwede akong makitulong as needed. And also, actually, for my case, I usually check with the student ano ba yung interest mo, ano ba yung passion mo in terms of food science, what is your interest to study, and definitely there will be questions that can be answered using metabolomics if you just identify a certain field of interest for you. So yun.
By the way, next week would be food metabolomics focused yung mga talks. So you better attend that as well.
Alright, thank you, Doc Fe and Doc Karen. I echo Doc Fe's sentiment na you have to be there next week dun sa food metabolomics session. It will be fun since the focus will be on coffee, and most of us, I think, are fond of coffee. Okay, so the next question is, hello, good morning. May I ask if the DLSU CIF can handle transgenic research like directed mutagenesis, generation of chimeric proteins, etc.? So again, for CIF related inquiries, I will send you a message. Okay. So next question would be from NM Panahon. Can we apply metabolomics and soil contamination assessment studies?
Okay. NM. So actually, if you talk about soil, I do have a project that is looking at soil metabolomics. But when we talk about kasi of metabolomics, we're looking at living systems. Mostly in terms of soil metabolomics, you're looking at like the metabolites of what is inside the soil. So ngayon, if we're looking at contaminants, soil contaminants, maybe not so much metabolomics, but yung untargeted approach of analysis using either GCMS and LCMS would be the approach that you would consider. So yung untargeted LCMS or GCMS can help you determine kung ano yung different contaminants that are present in the soil. Now, of course, if you are looking at kung ano ba yung effects of these contaminants to the microbiome, to the components, living organisms that are present in the soil, of course, definitely that can be done using.
All right, thank you, Doc Karen. So this is a question from the Google form. So this is from the Rename M. Alisada. I hope you're viewing us either from Zoom or from the Facebook Live stream. I am a senior high school student expressing an interest in the field of metabolomics. Wow. Sana all. Ayan. And I want to know what college courses can I take in line with this field. Thank you very much.
OMG! May bias kami ni Doc Karen kasi BS Chemistry graduates kami.
Analytical science background. So,
Oo,
ayun. But if you're looking more at the applications, definitely masyadong maraming different applications of metabolomics. So you can look at ano ba talaga yung area of passion mo in terms of research, and yun yung i-pursue mo.
May personally, I have a friends,
So,
maraming friends na they do
pwedeng
genomics.
analytical chemistry if you're more interested in terms of instrumentation and the application of metabolomics to different types of research, then siguro mas towards analytical chemistry yung pwede mo i-pursue. But if you're looking more towards specific applications like clinical research or like plant biology, you can consider life sciences courses and agricultural chemistry like Doc Fe. So yun.
In addition, remember that metabolomics is part of a systems biology approach. So marami kami ring collaborators na geneticists. So sila yung nag-i-interpret ng genomics components ng project. So you can still go to the omics field, pero not necessarily you're doing the metabolomics, but doing the genomics. And they're equally important when you're doing a holistic type of research.
All right, thank you, Doc Fe and Doc Karen, for the unbiased insights to the question. Okay, so again, you could go for chemistry related courses or genetics or systems biology. So actually, another we have an audience, Mr. Michael, who also chimed in. So sabi niya, go for BS in chemistry, BS in AgChem, agricultural chemistry, or BS in biochemistry. So that's his opinion on the topic. Okay. So next question would be from Miss Arlene Amador. Hope you're here. This is from the Google form also. Any suggestions for new plant metabolomics research?
Medyo wide yung coverage ng plant metabolomics. So again, this will depend kung ano ba talaga yung area of interest mo. As we've mentioned dun sa mga applications, for example, for Doc Fe, she has applications on rice. You can look also at applications in terms of like different other crops, in terms of transgenic research, in terms of like stress response, plant breeding. So, yun, super dae ng pwedeng applications ng metabolomics. So you just need to pin down kung ano talaga yung interest mo in terms of plant biology. So that will be my take on that.
Remember that metabolomics is a tool. Kaya ang dami-dami applications na pwede mong gawin. Kaya ang maganda doon, once you are familiar with the metabolomics, iba't ibang applications you can do. So my work is not limited to agricultural metabolomics or plant metabolomics. I can also do food or clinical metabolomics. So you just need to be expert on one thing. And then especially with tools like metabolomics, ang dami-dami mong pwedeng puntahan afterwards.
Thank you to our speakers for the answers. Another question is from Speedy Crisostomo. How can we use metabolomics in plant improvement programs?
Okay. Doc Fe, ikaw muna.
Breeding programs, crop improvement. As I mentioned in one of those projects that I presented, one of the informations that you get from metabolomics is the metabolites that can be considered as markers. No? So yun yung unique for that particular sample or unique because of a response to a particular stress, no? Na pwedeng gamitin information and translate it to the genomics component. So ito na yung pumapasok yung molecular breeding programs na ina-adapt ngayon. So based on, kasi yung present lang namin is yung present ko lang yung product na, yung grains, rice grains. Pero metabolomics, ang dami-daming applications in terms of salt response, drought response, and then they can also see there the metabolites that respond to that particular stress. And then look at those metabolite QTLs and use that for breeding programs or crop improvement programs. So madaming applications talaga if you want to use metabolomics for agricultural research.
All right. Thank you, Doc Fe. So, siguro last set of questions. This is another question from Tecton, Mr. Tecton or Ms. Tecton. Do we have an agricultural controlled room / area in the Philippines or in another country where we can control the weather so we can apply metabolomics in non-seasonal crops production? So agriculture controlled room or area.
Okay. That one, I'm not very familiar. Perhaps Doc Faye will answer that.
We have in the Philippines, I think international rice, in terms of rice to ha, kasi yung rice pwede mo talaga ilagay sa mga pots and then study its physiology, morphology, etc. There are glass houses, greenhouses, controlled temperature chambers that you can use to monitor yung effect ng environment, effect ng temperature, moisture, humidity, etc. You can do that. Yeah, the phytotron in Erie. I think Phil Rice also has facilities that similar. I'm not sure, pero tingin ko meron sila. You better check with Phil Rice. That is in terms of rice. But I know for growing or organically grown vegetables, meron na rin mga research ang mga vegetable big vegetable companies. They have mga temperature controlled environments or chambers na pwede rin. But I'm not sure whether it's open for for clients outside their company. Unlike research institutions na nag-offer sila ng collaborations or rent ng facilities nila. Pero meron, you don't have to go out of the country. Well, I'm speaking in behalf of Rice Rice Science kasi yun yung yung experience ko na we can conduct experiments like that. Pero kung bigger crops ang iniisip mo, hindi ako sure kung at what stage meron sa Philippines.
Alright, thank you po. This is the last question or actually set of questions. So connected sila. First this is from Miss Maria Adeline Antonio. What essential equipment / software should be available in metabolomic studies? And in connection to that, this is from Mr. Ian Gabriel Cuino. What are the technical requirements for capacitating labs to conduct research in this field? Okay, so yun po yung last set natin.
Okay. So, katulad nung discussion namin kanina ni Doc Fe, so depende kasi kung saan ka sa side in terms of like metabolomics. Are looking more towards NMR metabolomics. There are those that are more of mass spec based, but you have to consider in terms of NMR metabolomics, sensitivity is not as low in terms of like sensitivity. It's not as high as mass spec. So you might have to make use of more samples in order to detect some of these metabolites. And of course, you have to consider yung mga overlaps of signals for NMR. Mass spec based will be more, you have more flexibility in terms of like making your acquisitions more specific and more selective. So perhaps for basic metabolomics research, you would need either NMR or mass spec. You can have GCMS or LCMS or other separation techniques coupled to your mass spec. Kasi if you only have direct mass spec detection, ang problema doon, lahat lalabas at the same time, and they can co-interfere with each other. So usually there's a separation involved, be comprehensive list of metabolites and do untargeted analyses. Then you will have to go with high resolution LCMS and perhaps invest in also in at least a single quad GCMS for like those basic coverage of your metabolites that are polar and are volatile. Ano pa ba? And siguro in terms of software, marami namang available na freewares. I mean, there's a lot of currently available web-based, server-based softwares that you can use for data processing as well as for data analysis for metabolomics. So I think on that side, wala namang magiging problema. Pero yon, in terms of instrumentation, you would have to invest in those mass spec-based or NMR instrumentation. For NMR, it has to be high resolution. so that you'll be able to distinguish the metabolites kahit na merong konting overlaps. If you're looking at untargeted metabolomics for LCMS, it has also to be high resolution, like quadruple time of flight or bitrap. Um, if you're looking more for targeted applications, you're looking at specific compounds, then you can make use of those triple quads. So there's a lot of considerations in the instrumentation. So I hope that you will attend yung susunod nating session, and I'll cover more of those aspects dun sa susunod na session.
All right, thank you, Doc Karen. And
Oh, by the
thank you
way,
din.
I
Sige po,
think
sige po.
one of the biggest concerns also in terms of like of these instruments is that you need to have, of course, stable power sources, yung gas requirements and all. If you're more interested, if you want to know more about those requirements, I think you can talk to the people of CIF, and they will be able to provide you kung ano ba yung mga requirements that the suppliers are actually ask of them. So yun, kailangan mo, especially in the Philippine settings, madalas ang brownout, you have to consider investing in UPS generators, yun. So ayun, okay, Chris, sorry.
And thank you po, Doc Karen and Doc Fe, for your very insightful answers. So, in connection to the power issue, we've been also experiencing that here sa CIF. So, nung taal last year, it really was a big problem, and then yung intermittent na mga power failure. So, please check your facility kung ano yung power situation niyo dyan. Okay? So, ayun, thank you so much to our speakers and to our audience who participated and who asked questions. And that wraps up our open forum. We have posted the link. I think Alona, one of our staff, posted the link to the evaluation form in the chat box of Zoom and in the comment section for the Facebook live stream as well. The cutoff for sending the evaluation form, take note, is up to 11:59 p.m. only tonight. If you wish to have an e-certificate, you can do so So again, before formally ending the webinar, let's call on again our director, Dr. Camacho, for the closing remarks.
Okay, so today we learned a lot about metabolomics and its applications in plant metabolomics, and we hope that the knowledge you gained today will not simply be a cerebral exercise on you, but we hope to transform that knowledge into real action in doing actual research, in discovering new things. If before we are limited by knowledge and instrumentation, now we are very much capable knowing that instrumentation is not a hindrance anymore, as we at DLSU CIF can help your research needs. Partner with us and contact us for more discussions on research projects. Our country needs more researchers. We need you to indulge in research. There's a lot of research things to do, and we need more catching up as a nation with respect to knowledge creation and research activities. So together with DOST, help us build a research culture in the country by dedicating your career in research. And mind you, it's very rewarding. So again, thank you so much for attending this session, and we thank you, Dr. Lacerna and Dr. Kalingasyon, for sharing their expertise. We will continue the sessions on March 13 and March 19, mark the dates, for more details about how to do metabolomics and its applications in food and clinical metabolomics. Again, thank you. Keep safe always and have a nice day.
All right, thank you, Sir Drex, for the message. Ayun, so hopefully we can inspire you to become researchers as well and contribute to our country's output. Okay? So from all of us, before we end, we would like to request you to turn on your cameras for a Zoom fee if you're okay with you're if you're okay with that. So this is for documentation purposes. So again, I would like to request you to open your cameras, please, for a quick screenshot or Zoom fee, quote unquote. So magka-countdown tayo. Merong 4 pages, so this may take a while, okay? So again, let's start with the first screenshot. 1, 2, 3. And then second one. Okay. So starting in 1, 2, 3. So marami-rami tayo. Third page. Okay. Again. 1, 2, 3. Oops. And then the last one. For the fourth page. 1, 2, and 3. All right. So, ox na tayo. Thank you so much. From all of us here in the DLSU-CIF, we hope you enjoy the webinar and we look forward to seeing you again in the next 2 sessions. That would be next Saturday, March 13, and next Friday, March 19, okay, for the food and clinical metabolomic sessions. For those who weren't able to register, we will be opening the reg form again, the Google form, in a short while. So please follow the DLSU-CIF page for updates and future webinar schedules. Take care and continue to stay safe and be healthy. Bye.